So we wrote critical review for Lancet Neurology. Can you hear? You cannot hear? No. I'm sorry. Can you hear now?
Yes.
That's very interesting. Okay. So thank you very much. So Ben and Vincenzo and I wrote a paper, and in that paper, actually attached to this handout, Ben really suggested we should mention that guideline renewal updating is necessary. So obviously that was very important suggestions. So we wrote down, and then September 2014, we had excellent summit meeting held by Eric Sorensen in Minneapolis. At that time, leadership decided we really need this guideline renewal and update. So I volunteered to organize this meeting because we did Tarrytown House meeting to 2011. So organizing committee, advisory board meeting, board actually committees interchangeably. They helped us to build up this workshop. So also applied NINDS applications for the meeting, and obviously after fortunately grant funded, Robin, and who is not able to come here, and also Amelie helped to build and correct right workshop. So that was very, very helpful. Particularly, Amelie not only helped these guidelines, also helped to lead Group 1 for preclinical studies. I'd like to appreciate Amelie. I don't know where she is. Thank you so much. Really, she needs hands. So again, we had a lot of funding. And when I applied NIH grant, and I also asked the ALS Association, MDA, and also British MNDA, and they said, fine, sounds important, but we'll give you funds if NIH approved. So we're kind of anxious time and waited. Meantime, a lot of people are funded, starting from Adams Foundation. Also, I have to mention private foundation, Anthony Sinatra, one of our patients. With the foundation, they have almost now 10, 12 years softball tournament and give us research funds. This year, also, they funded this meeting. So really appreciate it. If you look at all those names, pharmaceutical companies, and if you look at also not just United States, Canada, Japan, and Europe, so many people are funded. And so this is, I say, really ALS community funded this. You can look at all detail in the handout. So really appreciate it, their support. So if you look at also attendees, you can see many different disciplines. Basic scientists, biostatistics, clinicians, trialists, and pharmaceuticals, young investigators. So again, representing our community. If you look at the countries, many different, USA, Canada, Europe, Japan, Australia, and all those different countries coming to this meeting. So again, we really appreciate it. So I have to say, at the kickoff first sessions, starting from Marion Haberkamp from EMA, and also Dr. Ichimaru is from PMDA, Japan, and also originally Dr. Farkas, Ronald Farkas was supposed to be here, but he became unable to come. He was working actively on the project, and also he's working very hard for this area. So Bob Miller made a special arrangement and most kindly tight schedule with some Bryan came to give us a talk. So once again, we appreciate it. Then we have two sessions of learning. We have Theodore and Carl Kiberts and Andrews, different field, epilepsy, Parkinson's disease, cancer, we can learn. This evening session will be moderated by Bob Miller and Nigel Lee. And tomorrow evening sessions, and Donald Jones from Biogen, and also Andy from Cytokinetics, and Mike from KNOP, and Bernard from Discovery Therapeutics. So really appreciate, and this session will be moderated by Merit and also John Glass. But most important thing is this is the guidelines. So I thought that this should be evidence-based. And I said that this has to be evidence-based, And then Mike raised the question, cannot be evidence-based because we don't have enough evidence. And also we did some survey monkeys. You solicited some question on what's the most important things. And we build up those questionnaire. And Ettore and also Mike raised, this is a little bit more redundant, disorganized. So they took research question very well organized. So really appreciate people such as Amelie and Michael Swash who become unable to come this time, but that's group one, and also Kathy and Vincenzo and Leonard and Eric and Chris and Jeff is doing different sections. Also Eric, Adriano, and group three, Mike and Martin and Marcus. Group three, and then 4As, Jinsey and James, and Angela and Philip, of course. And again, very importantly, Eric McLean was helped for biostatistics. They had so worked hard, and of course with everyone else, and I think they should have some our hands. Really appreciate their effort. I must say this meeting is not end of their effort. Almost this is the beginning. So really they have to continue to work with everyone. So again, we have a number of pals, and Becky Kidd and Rick Isaacs and Ted sorry, Tassero and Chris Simon. And so really, I want you to communicate and get your opinion there also in part of sessions, so part of groups guidelines. So really appreciate your, their coming. So most actually importantly, certainly I'm good into the logistics and so forth. It's relatively simple, raising funds, but scientific issues, Ben Brooks and also Bob Miller helped entire guidelines core group. And then again, you know, Gary Grontes from University of Kansas and AAN methodologist for those guidelines formation and so forth. Really appreciate their continued support. Once again, really appreciate their help. So when I heard Delphi method, I have to admit, what is Delphi method? Sounds good. So I quickly went to Google, looked at the Wikipedia. That sounds good. But time to time, I have anxiety reaction, and so I decided to have a vacation in Greece and went to the Temple of Apollo, and I asked, "What's happening?" I carried the paper and asked any recommendations, and there's a roar. It's kind of almost sounds like all Greek to me, and so I taped and came back to ask Georgia to interpret. And Georgia, of course, speaks fluent Greek. And it said, actually, tedious, but at end successful. So we got a very good oracle. And so my hope is premise of the workshop, we must be positive, encouraging, and facilitating. That language has to be those expression. Also, if we do it, and might as well do it right. So after the workshop, we must have more and even better clinical trials in ALS. And also we should have a more international collaboration. This is the key. We have so many different nationalities and working together for same cause. And also we should have a stronger collaboration between basic scientists and clinical trialists in ALS. So lastly, I volunteered because I have a secret weapon. And Georgia did the last meeting organized, so she's experienced. But we are not meeting organizer and professional. We are research group. So she single-handedly everything so well, and this is the product of what she did. So really appreciate it. And so many times she had psychological stress and so forth. So even she decided to go to psychological PhD from this fall. So now she cannot do everything. Now we have a research team. John, who know John, he is actually a research coordinator for ALS Cosmos Studies now Arrest ALS project, and also Brittany is another research assistant, and Tara is a social worker student at Columbia, joined to help this meeting. So once again, really appreciate their help. And so this is it. So we are going to start our first session. Actually, not the session, but Georgia is going to give some housekeeping issues.
Hi, everyone. I'm going to speak really fast. So if you need interpretation, just come find me and ask questions. The program has changed slightly for today. You'll find an updated version on each of your tables. Also, the online handout has the updated program. The timing has changed just a little bit for this morning and afternoon. Table in the front over here is reserved for upcoming speakers. If you see your name coming up, please move to this table to sit down so you can quickly transition. For the speakers moderators, you will have a lighted timer. It's up here. Green means you have plenty of time, yellow means you have to sum up, and red means you've run out of time. For the speakers who have 8 minutes of presenting time and 10-minute discussion time for a total of 18 minutes, you'll see the yellow light come on when you have 3 minutes of your speaking time left. For speakers who have 25 minutes of presenting time and 5 minutes discussion time, you'll see the yellow light come on at 20 minutes. You will see microphones posted at the side of the rooms, here, and one up front. If you can't get to one, please tell one of the staff to bring you a portable mic. The mic at the podium and right here and the front will always be on, and the moderator mics and stand-up mics will require you to press a button or switch it on and off. There's power at every table for laptops. If there's not enough room for everyone to be plugged in at the same time, please share. For the webinar, it's currently happening right now. We're streaming live. There are 142 people registered. The entire workshop will be audio streamed and recorded, and registrants will be able to hear everything being spoken. For this reason, we ask everyone to state their name before speaking, and the recording will be made available to everyone after the workshop. St. Patrick's Day today, we'll have a treat between 7 and 7:30 in this room, so come back early before the evening program. And then any other issues, please ask me or the staff members. Okay, thank you.
So as you know, early house guideline meetings organized by Ted Munsat and his WFN groups. And I'd like to ask Wally Bradley to present talking about Ted Munsat. And I think it's really, he started so much good things for us. So Wally is my mentor for ALS, and also, of course, Professor Emeritus and Chairman and at the University of Miami. Also, of course, everyone knows he's an ALS expert. So Wally, please. Thank you.
So happy St. Patrick's Day. First slide. Can we get there? On here? Okay. Good. Ted Munsat, I think almost everyone in this room must know. He was born in Rutland, Vermont. He did his training at Columbia Presbyterian. He went to UCLA for almost 10 years and then came back to the Northeast. Ted was the quintessential ALS neurologist. He wrote many articles and books and won many awards, including several related to ALS. He was chairman of the Department of Neurology in Tufts, and he attracted me to come to the United States. And more important from this meeting's point of view is that he was chairman of the World Federation of Neurology subcommittee on motor neuron disease. And it was in that capacity that he organized the first of these symposia, which was held in El Escorial. The preface to that, the paper that came out of that, was that because clinicians and scientists worldwide are in need of precise diagnostic criteria to apply in order for the studies to proceed, that workshop was first organized. And that's really still what we have passed on to these further meetings in Ailey House. This is the cup that we got in El Escorial, and though we call it the first of these symposia, it in fact was, as you can see, the fourth reunion of ALS. I don't know anything about the other ones, and I'm not sure that anyone now in this room knows anything about the other three that were obviously held in, I would guess, in Spain. The criteria that came out of that then led to further discussion, further realization that we needed more clinical trials, that then led to a meeting here in Ailey House that considered the role of therapeutic trials, the role of industry, and as Hiroshi says, there's a further meeting. And that in red is what is the highlight of what was the goal of the workshop that was held here in 1998. And here are just a few of the pictures, some of them thanks to Patricia Anders, who sent me some that I had. You will see people around in this meeting room who now look a little older. This is Ted, this is Hiroshi here. Somehow the years do seem to pass on, and but we're still here, and that's perhaps the most important part about it. Here's Ted with his wife Carla, my wife Jean, Ted with King Engel and Vala Ashkenaz. Ted was a great sailor and loved sailing, and that I think is what I should leave you with. I have also one other announcement to make. Some of you may know that John Walton, Lord Walton of Detchant, my old mentor, sadly collapsed in the House of Lords a week ago. He's now in hospital in his hometown of Newcastle, and I have already sent from all of the members of this meeting here our best wishes for him. So thank you.
So some of you know we are doing guidelines, clinical trial guidelines, but also simultaneously, in fact, started earlier, FDA new drug development guidance. And so people a little bit guidance and guidelines. And so we want to make sure we understand difference and similarity between them. So Lucie Bruijn is going to present what's the guideline and guidance, difference and importance and purpose. Thank you, Lucie.
So thank you very much for the opportunity to just quickly present this, and it really gives me the opportunity to thank many of you in the room here that when we approached you last April to see if you would work with us on this probably didn't realize quite how much work that would be. But it's been a tremendous effort from clinicians, patients, and many groups to build the FDA guidance. So I hope in these few slides I can help highlight what the differences are. And I also wanted to mention that really this was initiated by the ALS Association, although many ALS organizations are part of it. It's really a whole community effort, and it was in large part helped with the guidance and expertise of our consultants. Alison Durham is here in the room. Alison, I don't know if you can show where you are, so if there are particular questions, do connect with her. Dave Zook will be coming later as well. And we built on their expertise. They had done this for Duchenne's, which was also really how we tried to move forward on it in a similar way. So the guidance, and I'm not really going to read everything for you here, and it's difficult from this end, but the guidance is very different to the guidelines, although they are intentionally meant to align. And actually when Hiroshi came to me asking for funding from the association for the guidelines meeting, I myself was a bit confused, and I thought, what is the difference here? And realized that actually if we don't do this in tandem, it wouldn't make much sense. And in fact, I think all the work that has been done by many of you in the room to build the guidance document has helped set the stage for discussions today, and we definitely hope the two will align very well. So the guidance is something that the FDA sometimes themselves initiate for diseases, but in many cases they don't have the funds necessarily for all diseases. I have to say that's changed a bit for ALS. There's much more interest and attention, I think, from the FDA, and they are internally working towards a similar effort. So these guidances have actually been accelerated because of those synergies. The guidance is really a tool to provide critical guidance for clinical trial design, implementation, and standards. So it's the FDA that finally comes out with that document, but we can influence what that document looks like. It's a roadmap to navigate regulatory processes. So they are, as I say, usually developed by FDA, but in this case we were able to also, with the Ice Bucket funds, initiate it so that it would speed up more quickly. These guidances can take a long time. And these guidance materials also include clinical trial outcome measures and in-depth discussions of things like benefit and risk, some of the things that might not be as strongly included in a guidelines effort. So the guidelines, as many of you are well aware of because you've been engaged in this, are really tools to develop best practices, can be used by the researchers and industry, and are really more describing the way to conduct clinical trials, but are not really directly going to impact regulatory processes, although I think hand in hand they can provide some influence. As you see, both are going to cover many of the same topics. I think importantly, just to give you a sense of the guidance effort, how that was structured, it was both Hiroshi and I are co-chairs on this, and I helped guide our public policy team who the players might be to engage around this. And so it's very much engaging the ALS community. The caregivers and the patients are on an advisory committee. Each of the advisory committee members of the patient committee are also a member on each of the working groups. So they've been very integrated in the discussions and the process. We identified these as being the key working group areas: policy, benefit, risk, biomarkers, natural history, diagnosis, clinical trials, outcome measures, and frontal temporal dementia. These working groups have been incredibly busy and active and have set the stage for this document through much discussion and trying to align different views. The draft guidance has in fact in first draft format been provided to the FDA. The FDA have been very engaged with us actually with this. There was a meeting in person with Janet Woodcock. They're very, very on board and quite excited about this. There's obviously a very active patient community who've been wanting to see things move faster, and I think this guidance and these relationships have been very helpful all the way around. So the intention was then, because this meeting was planned as well, and in fact, I think it's tremendous that so much work has been done in advance of this meeting so that we're not coming in here and starting to talk about topics, that it's really very well structured, because the intention is that with quick order after this meeting, anything that is coming out through the guidelines will be informative for the final draft that will go out for public comment. So if you see here the time frames, it's quite tight time frames because we want to move this quite quickly. So that by the open comments, that open comment session will then be incorporated into the document that then we will collectively submit to the FDA. I think most of you may be familiar, but others not, that isn't a document that gets published by the FDA. The FDA takes that guidance document and from it develops their own guidance document, which then again goes out for public comment. So one of our big goals will be, and I think it's already read, so I have to go, but one of our big goals is going to be to communicate to all the stakeholders that what they finally see might not be quite what they had submitted, because this is obviously a document that but I think we can influence it hugely. So I hope that clarifies a little bit of the two efforts, that they are not meant to be completely distinct, but they have their own goals and hopefully will align. So thank you very much for the time.
So beginning sessions, Ben Brooks going to talk what's happening in this organization, guidelines development and so forth. So Ben, please.
Good. The background for this comes from the fact that the FDA had a public meeting on February 25th, 2013, that led to the whole session of how the United States and many people felt about ALS being on a webinar that's available. All these slides are available through a link that you will have. So all the slides from the conference also are available to you, so you can get these links. There are 269 open comments, and the FDA responded to these. We've had two public seminars with respect to this, one in Bloomington and then again in Brussels. The Europeans were ahead of us in terms of looking at some of these issues. There are substantive differences between the approaches and recommendations between the United States and Europe. And there were, I think, 7 comments from European on the European outline. The Japanese have recently approved a new drug for ALS, and they have a similar website and presentation of their thoughts and processes on that, and we'll hear more about that today. The methods that we're going to go through today are primarily based on the different workshops, looking at the guidelines, and then coming through and revising them based on evidence that we have. What's going to happen immediately post-conference is that in the first week post-conference, you should have all your comments back to the group leaders. In the second week, the final post-conference version will be prepared for the finalized initial draft. That would be put online and reviewed. There'll be 4 weeks of comment. And then the final version, semi-final version of that will go out. And then open comments will be sent back to the leaders and to the Columbia website, and we will prepare a revision list based on those comments. Some of the revision recommendations will be accepted, some won't, and then the finalized revised document will go out for what's called the modified Delphi approach. All of the leader emails are online, but they're also a piece of paper on your table there to make sure you get your leaders' emails. This will be the main contact for everybody as we move through the process. The effort will be to create the first consensus-driven modified Delphi method approach to ALS. Here's the history of consensus guidelines. Thousands of clinical guidelines have been put in place, but only a few hundred these are for different diseases, have been put in place with respect to clinical trials guidelines. if we look at those that have been subjected to the Delphi approach, only 1 log less has actually got the Delphi approach for clinical guidelines, and the Delphi approach for clinical trials guidelines is so small that the NIH isn't putting up a graph of this kind of thing. However, this is the history of consensus ALS guidelines. We had the first Arley House in 1994. That was published in Journal of Neurological Sciences, but no longer on their website. We're going to talk to John England to try to get that back on their website so it's electronically available. Then Brinkman and a group of workers there did a second publication on outcome measures in ALS. So there were 2 substantive documents that came out of that guideline preparation that sort of drove and guided many clinical trials over the next few decades. And most recently, there was a trial published in European Journal of Neurology highlighting this guideline as one reason they went into the way they designed their trials, with an appropriate response from Dr. Ludolf, a negative response. So we then had the Arley House 1998 that was published in 1999. But the next level at the level of preclinical science came from the EAMNC and the ALS TDI with the Narden and Reisenberg conferences. And then there was a stem cell network specifically dedicated to ALS with respect to guidelines for stem cell trials. And then at the second Tarrytown conference, there were 2 publications trying to look at the infrastructure and where we should go in terms of the guidelines. So my last comment to you is many people have identified that this is a webinar, so make sure that when you put your finger in your ear, that's okay, but don't put it in your nose because people are watching, and we'll want to make sure that we're all on good behavior. Thank you very much.
So now we are going to start our kickoff sessions. I would like to invite Gen and also Stan to come to moderate. And so this is the session from regulatory agency from Europe and Japan and United States.
Good morning, ladies and gentlemen. I from Japan and Dr. Stanley Appel from Houston will chair this session. This session is, as Lucie already told, very well organized to represent the whole meeting of this meeting. And as you know, understandings of the genetic and biological backgrounds of ALS very much improved since the last guideline meeting over here almost 16 years ago. So now I think it's a good time to realize and remodel the guideline in a very historical place. And in the first session, 3 representative speakers will present representing the regulatory agencies of FDA of the United States, PMDA of Japan, and EMA of New York. And probably I think all these 3 talks will be very, very informative and very suggestive for thinking about the guideline for drug development for ALS. So for the first talk and the last talk by Professor Haberkamp and last talk by Wilson Bryan will be chaired by Dr. Stanley Appel. And the second talk will be presented by Ichimaru from Japan, our chair. So let's start the first talk. So I want to introduce, first before I start, I just want to add to Jen's comment. It is critically important that we start off with recognizing how valuable the input from the agencies in all our countries are. Because we can get to the starting line, but we can't start without their approval. And so having them involved in this consensus conference is critically important so that we're all on the same wavelength, we're all starting at the same time, and we have an opportunity to bring fabulous new drugs to the patients. So our first speaker, representing the European Medicine Agency, is Dr. Marion Haberkamp, who trained as an internist in Bonn and Cologne, then joined the Federal Institute of Drugs and Medical Devices in Germany, and has been involved in neurology and psychiatry more recently, and she helped write the guidelines for ALS. So, Dr. Haberkamp, welcome. Look forward to your comments.
Yes, thank you. Good morning, everybody. And it's really a great honor for me to be invited to this important meeting in this historical place, and a pleasure to present to you the key issues of the European guideline on clinical investigations of medicinal products for the treatment of ALS. And well, this is my disclaimer. I'm a senior clinical assessor at the German Medicines Agency in Bonn, and I'm also involved as an expert in the Scientific Advice Working Party and the CNS Working Party at the European Medicines Agency, and I was responsible for the draft of this guideline. I have no conflicts of interest. We will learn in the following 2.5 days a lot about clinical and non-clinical science, but my talk focuses on regulatory science, and hopefully these parts are not too these fields are not too much apart, since our joint interest should be to really foster development programs in this seriously debilitating disease, and we urgently need new medicines, and all have to work together. I will start with a short history of the guidance document, which replaces the old points to consider document from 1998. Interestingly, this one came out around the same time at your Miller and Brooks consensus guideline on clinical trials in ALS. And the history, the timelines were that in August 2013, we released our draft guidance document after lots of internal rounds. Finally, we had the draft in 2013, and this is how it is usually done for half a year for external consultation. In your handout, you have the draft guidance, not the final guidance, and we had lots of work up in between, so that is no longer up to date, and I will bring to your attention those key issues which we changed. And after the external consultation in January 2014, we needed almost 2 years to finally draft the guidance document, because so much knowledge was building up and publications were coming out. And the final guideline, however, has been published since December 2015 on the EMA website, where you can see it. We also published an overview of comments document where we listed all the external comments, mostly from industry, learned societies, but also academia. And you can find, if you are interested in this document, which is also on the website, the reasons what we took on board, what we modified, and also the reasons why we rejected some of the comments. Well, what has been additionally taken into account, most up-to-date research data and information from clinical trials in ALS as far as they came to our knowledge, mostly via scientific advice or protocol assistances at the EMA. We try to do a constant literature update, including reviews approximately till the mid of 2015. So the guidance drafting works like that. So we can never be comprehensive, and there are lots of publications out there which might be missing in the list, which you might notice. So we were also aware of the approval of edaravone, the Radicat, a free radical scavenger in Japan in the mid of last year, and we will hear about that in the following talk. What has been changed in the guideline? Well, the main requirements for the treatment of ALS with respect to diagnostic criteria, inclusion criteria, trial designs and endpoints were revised. The guidance focuses on treatments for disease, modifying treatments for ALS, but also some guidance was taking on board for symptomatic treatments, since companies were coming to us developing those compounds working mostly on the muscle and on function. But non-specific symptomatic treatments, although important for the patients, like drooling or a pseudobulbar effect, are not within the scope of the guidelines. And there's a sentence in the guideline that primary lateral sclerosis with pure upper motor neuron development and PMA with pure lower motor neuron involvement are not within the scope of the guidelines, since the course of these diseases might be quite distinct. However, guidance might also be needed here, and patients might be needed to be included into studies. So, and that is what I want to state right from the beginning. It is a guidance document, and the guidance is never set in stone. So it might quite soon need already updating and amending according to the emerging knowledge. So in the introduction, we briefly summarized the epidemiology of the disease, which is quite well known to you. So reported incidence varies across regions, 0.3 to 2.5 per 100,000 inhabitants. The etiology and the reasons, pathophysiology of ALS is not fully clarified, unfortunately, so complex interaction between genetic and molecular pathways is assumed. We traditionally have the division between sporadic and familial or hereditary ALS. However, a growing number of ALS-associated genes have been identified recently, and especially the identification of the C9orf21 repeat expansion in patients without a family history of ALS has challenged this traditional division between familial and sporadic ALS. And there is, of course, the overlap between other neurodegenerative diseases, the frontotemporal dementia, which we have to take into account. Median survival time is unfortunately still very low, 2 to 3 years. They are longer disease courses, and it takes still around 15 months worldwide to establish the diagnosis, despite the refined diagnostic criteria we do have. And the only approved treatment in Europe so far is riluzole for disease modification. So we definitely and desperately need more treatments for this disease. So what should be the main goals and study objectives? Of course, according to this, to increase survival, but also a delay of disease progression. Some guidance is given on improvement of symptoms, it's already mentioned, muscle strength and related function. And of course, the ultimate goal would be primary prevention, and this might be the future, and we are not there yet, and a proper guidance cannot be given. So we just mention and briefly hint out that this might be future. And the improvement of quality of life, of course, for the patients may be an important secondary goal in trials. So the guideline also addresses this. So what is about patient characteristics and inclusion criteria in the guideline? So what is new to the draft is that we now accept definite, probably, and possible ALS for the trials. Since we were convinced that possible ALS is convincingly sure of developing ALS in the future, so we were convinced that those patients could be included into trials, and the basis for this is still the revised LS-Corial criteria. The diagnosis is mainly clinical with some electrophysiology, and we are aware that refined criteria might pretty much emerge in the future. There seems to be activities out there, and I mentioned the publications from Ludolf and Augusta. The AVAI electrodiagnostic criteria increase the sensitivity and have a good specificity. However, the clinical usefulness might not be established everywhere. So that's why we are cautious a little bit here. And we are aware that those criteria categorize the diagnostic certainty, but we have extreme heterogeneity among the patient populations, and that's the difficulty for the trials. How can we define homogeneous populations where we can really show an effect, and how is this representative for the whole population? And here, validated prognostic models may help to stratify the study population, and this might be helpful if it is a good concept. So this is the summary of the revised LS-Corial criteria. I don't go into details here. You are, of course, all aware of that. What do we change in the exclusion criteria? Well, excluded should, of course, be other causes of neuromuscular weakness. That's clear. We have no longer what was in the old guidance document, the dementia as an absolute exclusion criteria, because ALS is associated with frontotemporal dementia in about 14% of incident cases, and cognitive impairment is an important feature of ALS, and we, almost 30% or even more have cognitive impairment, and we should be aware of that, and this feature needs to be studied, and patients with these features need to be included in the studies. We have a chapter on therapeutic efficacy measures. What is important here? Well, of course, survival and the time to failure analysis, and we stress that if survival is not a primary endpoint, as it usually should be in disease-modifying treatments, it should at least be secondary. And of course, survival data may be confounded by end-of-life measurements like ventilation strategies, and use may therefore, of course, be made of a composite endpoint of time to event endpoint, or recording time to death, and tracheostomy, and permanent continuous ventilator dependence. Functional measurements are, of course, of key importance. We mentioned as an example, and the most and best studied and validated example, the ALS functional rating scale and its revision. However, we also are open to other functional scales as far as they are justified and validated. So that's a guidance, so we cannot be too prescriptive here. And we mention, of course, muscle strength as a third important endpoint. Usually a secondary endpoint, but it might be primary when it comes to disease modification, and it's definitely important in phase 2 trials. Respiratory function should always be measured in ALS trials, and there are examples mentioned what should be measured here: the vital capacity, forced vital capacity, and the SNP, which are informative. But blood gas analysis might also be informative. That's not in the guidance, but that's for sure. And assessment of health-related quality of life is important, and we have generic and disease-specific scales here that can be used, and that's important to a secondary endpoint. And of course, we have also global measures where we have anchor points we can use, and additional endpoints which are newly mentioned, that is electrophysiological assessment, Munich, staging of disease progression, and cognitive functioning. Now we come to studies. Exploratory studies are important. The usual way how it is done, that we have a standard approach, phase 1 studies to find the safe dose, followed by phase 2 studies to determine the biologic activity. The use of motor and respiratory measures in phase 2 might help to a smaller sample size and shorter study duration, but we have to be careful if this really foresees the outcome in phase 3 studies. Therefore, important is the use of PK and PD data to establish dose response and the demonstration of target engagement. Some modifications are acceptable, so that it might be not always be possible to start in healthy volunteers if the mechanism of action is not established in healthy volunteers, so we might start in patients in phase 1. And preclinical efficacy testing is important, and the model should be carefully chosen. Here some development is already underway. So the guidelines is divided with respect to clinical efficacy studies into studies for disease modification and symptomatic treatment. That's why companies come to us with these claims, and we felt we have to give guidance on that. However, in the future, it might be more guided by the patient population. But for now, it is divided into these specific aspects. So what do we need? Parallel double-blind randomized placebo-controlled trials, and historical controls are not acceptable. That is stated in the guidelines due to the confounding effects and the difficulty to establish a causal relationship with the historical controls. And since riluzole is approved for disease modification, placebo-controlled trials would most be for ethical reasons be in an add-on design or for future upcoming treatment. So please, this can still be a placebo-controlled trial if you do it in add-on to riluzole, as far as the mechanism of action allows for that. And we can, of course, think about superiority trials to riluzole or an upcoming disease-modifying treatment once it is approved. Non-inferiority trials are not acceptable due to assay sensitivity reasons. The study duration depends on the expected date of progression, which in turn depends on the population included, and usually is 12 to 18 months in disease modification. So now it comes to efficacy endpoints. What is the key change to the draft guidance in your outhand is that we the main point, and that what was the main point for discussion during external consultation and among ourselves was the two co-primary endpoint requests from the domains of survival function and muscle strength. And the guideline now states that either time to death or function or both can be used. And for proof of efficacy, a clear and significant effect in one domain and a trend in the other should be sufficient. And ideally, a response criterion that could be considered clinically meaningful should be defined a priori if it is possible. And there are also alternatives mentioned. For example, a time to event endpoint with the event defined as death or predefined deterioration on a functional scale, the ALS, a functional rating scale, or even a composite endpoint of survival and functioning, and there are examples for that in literature. However, what we stress is that the choice of the primary endpoint should not lead to insufficient data for assessing the effect on survival, because this establishes the clinical meaningfulness in the disease modification claim. We were faced with uncertainties of the rates of change in ALS, a functional rating scale, the slope analysis, and this slide enumerates some of those uncertainties. So what is considered clinically meaningful here, 20 to 25% change, at least somewhat clinically meaningful. What does somewhat mean? It is clear that it coincides with the disease progression over time. However, this assumes the linearity of the disease course, which might not be the case. It might be rather curvilinear, and there's of course a concern in the presence of early withdrawals or deaths in measuring slope analysis. So a risk of overestimation of treatment effect, and we have to carefully look at those data, and therefore we recommend that it should be related to a delay in time to an important clinical event, for example, assisted ventilation or death. Well, clinical efficacy studies in symptomatic treatments, there's no compound approved yet here, so we need two-arm parallel group double-blind randomized placebo-controlled studies, and superiority to placebo should be shown. Add-on in design on top of riluzole, of course, is possible, and stratification can be undertaken. An effect on disease progression should still be measured. Depending on the mechanism of action, study duration of 3 to 6 months duration could be sufficient, so shorter than in disease modification treatments, and safety data of over 12 months should be collected, of course. Muscle strength and function are important endpoints and should show consistency in effects. And one should be selected as a primary and the other as a secondary endpoint. That's what states the guideline. And an effect on disease progression and survival should still be measured, also in symptomatic treatments. However, this time to exclude a negative effect of treatment. There are short chapters on studies in special populations. I would not go into detail here for the sake of time. We say that children and adolescents, if it occurs in young patients, it's difficult to establish studies here. So patients might be included into the adult studies, and with respect to older patients, then patients are getting increasingly older to better standards of care. An effort should be made to recruit patients in older age ranges over 65 years and where possible over 75 years, 75 years of age. Safety evaluations according to the ICH E9 note for guidance are recommended, and there are of course specific safety aspects that should be carefully measured and collected in this population: thromboembolic events, aspiration, pneumonia, and malnutrition, and this interferes all often with treatment. So I already would like to give, towards the end of my talk, an outlook what might be important for the future, a revision of existing diagnostic criteria, which is already underway, the development of a staging system, which might be helpful, neurophysiology, which is increasingly standardized, and especially in phase 2, but also in phase 3 is important, cognitive endpoints is important. What can we recommend here? Is there anything validated? It was not in 2015, but the literature, I went to the literature, there seems to be the ECAS, validated scales that can be used. Genetics is important. All patients might be tested, or at least DNA should be obtained from the patients. And to my personal opinion, biomarkers are especially important. And there are all kinds of biofluid-derived neuroimaging or electrophysiological biomarkers out there. And their use can be either diagnostic, predictive, prognostic, or for enrichment. And that might be especially intriguing. However, for now, we have no validation for all of these specific purposes. So from a clinical trial perspective, we were of the opinion that the existing criteria, the EC criteria with minor modifications, remain still fit for purpose. So what can be offered in case new information is coming up and the guidance is it's a slow process, a redundant process. So early involvement of the regulatory bodies, either through scientific advice and protocol assistance, European level or national level. We also have a tool of CHMP qualification opinion on the acceptability of specific use of proposed methods based on data assessments, for example, the use of a biomarker or a certain trial method in a disease. And a step further down, we have a CHMP qualification advices, which is based on less complete data, where the methodology is developed. And we also already had in other fields joint efforts with the FTA, which were especially fruitful. So lots of interaction between the agencies is already going on. I want to close with a high-level overview on early access tools once it comes to marketing authorization, to bring this to your attention. We already have the conditional marketing authorization, which is an early approval scheme on the basis of less complete data, and we have the accelerated assessment procedure during centralized procedure when there's an unmet urgent need to shorten the assessment time from 210 to 150 days. There's ongoing revision on the guidance of those procedures, and we also have two emerging new procedures: the priority, the PRIME scheme, which is a new scheme to reinforce early dialogue and regulatory support for innovative medicines, and which are also thought for accelerated approval. And we have a pilot running on the adaptive pathway, an adaptive pathway project, which is a more open dialogue between all kinds of bodies, also HTA bodies, healthcare decision makers, and a very open platform. So it might be all documents on that you can all find on the EMA website, and the message, the take-home message for that is just, it might be useful also in planning for marketing authorizations to talk to the regulatory bodies very early. And I want to thank you now for your attention. Vielen herzlichen Dank. These are my contact details. Yes, okay, go ahead.
My name is Ryuji Kaji from Japan, and I'm a member of MND study group of WFN. And my question here is, in case of edaravone approved in Japan, it only looked at function, so it didn't look at survival. But you mentioned that function, if you look at some function, you also have to see the survival also. But it takes a long time to see the survival. Do you have any recommendation or any suggestion? If you have functional improvement, but that could end up with a shorter survival. So in that case, what would you recommend?
Well, I'm pretty aware of some of the data on the edaravone dossier, and indeed this was a kind of why we were well, a little bit a deficit of the data. We thought, because we did not see any improvement in the harder outcome criteria, like survival, and that might be a concern. So the study duration should be long enough to see a little bit. So like 1.5 years probably, and that's what we usually recommend.
So for edaravone to be approved in Europe, then you might want to see that. Then.
Yes.
Okay, thank you.
You have to
Yeah, I can't hardly hear you, yes.
Is that better?
Yeah.
Bob Miller from San Francisco. You mentioned that historical controls are unacceptable. I wonder if you could comment. In Parkinson's, for example, where the rate of decline has changed over time, they have quite appropriately agreed not to use historical controls, but our FRS data in terms of slopes and things, although I know you had comments about slope, have been very constant over quite a few years. Might you comment about that?
Well, we generally, for all kinds of diseases, we don't accept in phase 3 as proof of concept historical controls because of the reasons really.
What about phase 2?
It might be acceptable in phase 2. But that's not in the guidance. It depends on what has to be shown, and that should be really discussed with the regulatory bodies in advance to run not into difficulties here. But for phase 3, we really have the feeling that due also to the evolving criteria, standard of care during regions, although it seems to be pretty much the same all around the world, but when there might be differences when it comes to ventilation strategies, for example, and that's why we have so many confounding effects. If this is a historical data from quite a long time ago, and then of course to establish the causal relationship, how you can do that in phase 3 in a pivotal trial if you don't have a comparator to which you can do that. So that's we had the feeling that that would not be the right way for the phase 3 trial design in this, even in ALS.
Yeah, my name is Leonard Vandenberg from Utrecht, the Netherlands, Europe. Hello, I'm here. Yep.
Yeah,
Hi, sorry. Yeah. I just have a short question. Thank you very much for presenting the guidelines in your presentation and for being here. I just have a question about how you establish these guidelines in Europe and how you involve people like us here in the audience in these guidelines. We discussed the guidelines in 2014 in our ALS group in Europe, and we submitted some adjustments in 2015, January. And so how have you used those, and what's the status of our comments?
Well, all those comments that came into the guidance, that's why we have the external consultation. It's on the website, and interested parties, industry, learned societies are of course invited to comment on that. And these comments are all collected and reviewed, and that's why I try to explain to you why we have this overview of comments document. So.
Okay.
Where we collect it and we review those comments, and we take some of them are really helpful, and we took them on board, and some of them were slightly modified that kicked off a thinking process, so we modified some of these inputs, and some of them even were rejected because we didn't feel that should be in the guidance document, or we were not quite there yet, and we explained it in the overview of comments document I mentioned, which you can find also on the website of the EMA. It was published in January this year, slightly after the guidance document, because it needs some time to, but you can find, hopefully your comments are in there, usually. So
they're definite now, the guidelines? Or?
Yeah, it's finished now. It has been published since December 2015 on the EMA website.
All right. Okay.
Yeah.
Thanks.
Thank you.
Yes. Eva Feldman, University of Michigan. Does your agency have an official stand on the use of sham surgeries as a placebo in stem cell trials for ALS?
No, not yet. So, but we might think of that, yes, of course, because this is emerging evidence that is coming, that is very new developments, and we don't have this in the guidance document yet.
Last question. Hi, Rick Bedlack from the defending champion Duke Blue Devils. So
last year, Dr.
I didn't get that.
I noticed when you talked about inclusion criteria, you didn't specify anything about disease duration. And I wondered if that was a conscious omission. Did you have a conversation about that and leave it out? Because in our guidelines, we said under 5 years. Most of the trials that we do now, we won't take patients who've had disease for more than 2 years. It's very frustrating for patients and also for investigators. The majority of the patients in my clinic don't qualify based on that criteria, and I'm not sure that we have good evidence to support that. So could you share some of the conversations around leaving that out?
Yes, well, we didn't specify that in the inclusion criteria for exactly these reasons, because it might be acceptable to have patients even on a longer disease duration in the trials we mentioned, and I did not go in all of the details. I picked out some of the key issues of the guidance, but if you go into the inclusion criteria sections, you see that we recommend stratification according to disease course, and that might be, of course, an important prognostic factor, that patients who have longer diseases have worse outcome, and that should be stratified, and that's what we mention in the guideline.
Thank you very much, Dr. Haberkamp. So we are going to second presentation by Dr. Ichimaru. I will briefly introduce Dr. Ichimaru. He's a director in the office of New Drug Section 3 in PMDA, and he's now engaged in the review process of many drugs for neurological diseases, including ALS. He was actually involved in the review process of edaravone. So today he's going to talk about the detailed procedure of the drug development of edaravone. Can you start?
Dr. Sobue, and thank you for your kind introduction. And good morning, everyone. I'm Katsuhiko Ichimaru from PMDA Japan. Today, I'd like to tell about PMDA's review on edaravone for ALS. This is disclaimer. This is today's outline. First, I'm going to tell you what edaravone is. This is important to understand dosage of edaravone for ALS. Next, I'm going to explain the results of clinical trials of edaravone with regard to ALS patients to consider the efficacy endpoint of these clinical trials for ALS. Finally, I'd like to talk about what we learned about ALS drug development from this edaravone. First topic, characteristics and indication of edaravone. This drug was developed by Mitsubishi Tanabe Pharma Corporation in Japan. The main characteristics of this drug is that it's a free radical scavenger, which means that the drug is a neuroprotective drug. The dose is applied through injection. At first, this drug was developed for the treatment of acute ischemic stroke. This indication was approved in 2001. In 2002, we issued a warning via healthcare professional letters of emergent safety communications about contraindication to patients with severe renal impairment. This caution is also considered in the indication of for ALS. In 2015, edaravone was approved for ALS in Japan. The indication for stroke is mainly focused on the improvement of neurological symptoms and LDL. In ALS, the indication is focused on the progression of ALS in functional disorder. Precaution on the ALS indication includes Japan, ALS severity classification, grade 4 or worse, and percentage free and 70%. This is the Japanese ALS severity classification score. Grade 4 is respiratory insufficiency or other verbal symptoms. Grade 5 is tracheostomy and using mechanical ventilation. Renal failure is one of the contraindications of this drug. Normally, serum creatinine level and estimated GFR are used to evaluate renal function. In ALS patients, muscle volume decreases significantly in advanced phase. This means that serum creatinine level is insufficient to evaluate renal function. So we adapted cystatin C to compensate. These considerations are described in package insert. Let's move on the dosage and administration. Post-stroke, this drug is indicated for the acute phase of ischemic stroke, that is within 24 hours after the onset of the stroke. The drug is administered twice a day and for up to 14 days. For ALS, this drug is used throughout its course. Administration in ALS is once a day, cyclically. For the first cycle, the drug is administered for 14 consecutive days, followed 14 days sedation. For the second and after cycles, the drug is administered for 10 out of 40 days, followed by 14 days sedation. The first cycle is similar to the dosage in stroke. Feasibility of clinical settings were considered in the second and after cycles. This is an example of second and after cycle administration schedule. To summarize the first section, the dosage was inherited and expanded from the indication of stroke. The safety monitoring methods for renal failure were modified in ALS. Move on the second topic. Let's look into the efficacy endpoints. Before looking at the efficacy endpoints, let's overview the clinical data package of edaravone for ALS. This is the clinical data package. Mainly, the efficacy of edaravone was tested in mild to moderate severity ALS patients with a score of 1 or 2. The efficacy of moderate severity ALS patients with a score of 3 was evaluated in another study named MCI 1618 study. MCI 186-16 study was the first confirmatory trial, but it failed. Afterwards, MCI 186-19 study was conducted with enrichment and it met succeed. This is a design of pivotal study MCI 186-19 study. It is a placebo-controlled, randomized, double-blinded, parallel group comparison study with sample size of 128. Ultimately included 137 patients. Efficacy was evaluated after 6 cycles of edaravone administration, with 1 cycle being 4 weeks. So, evaluation period was approximately 6 months. The first cycle treatment consisted of 2 weeks daily treatment period and a 2-week rest period. From cycle 2 to 6 of treatment, i.e. a total of 5 cycles. 1 cycle consisted of a 2-week treatment period in which 10 doses were administered, and a 2 weeks rest period. This is the same as the indication of ALS. In Japan, Nidazole was approved in 1999. ALS patients in Japan are given financial aids for ALS treatment provided by the government. This support might explain the prevalence of tracheostomy and mechanical ventilation in Japan. Then, the proportion of patients who use mechanical ventilation is more common in Japan than in Western countries, such as US, UK, and other European countries. Taking these facts into consideration, we need to consider about equivalence of time to death and time to intubation, tracheostomy, as efficacy endpoint in Japan. These are efficacy endpoints of pivotal study of MCI 186-19. ALS-FRS is the primary endpoint. Secondary endpoints include survival and other clinical events, percent efficacy, modified knowledge scale, ALDA-40, and so on, etc. The results of primary endpoints is 2.49 in ALS-FRS score over 6 months. This difference is equivalent to delaying the progression of ALS-FRS for 2 months in 6 months period. Other endpoints, results of other functional endpoints, modified knowledge scale and percent efficacy and QOL, ALDA-40, were consistent with ALS-FRS. These slides demonstrated the progression of ALS-FRS score in the extension phase. Difference between the placebo arm and the edaravone arm remained at the end of the extension phase in spite of administering edaravone administration to all patients. When we looked at these results about function, we need to consider the background of subjects who are mild to moderate severity ALS patients. Looking at another 40 score, the results are consistent with those of ALS-FRS. Let's move on to the deaths and other clinical events. During double-blind phase, which lasted 6 months, there were no deaths and only a few clinical events. However, during the extended phase, there were several deaths and a variety of clinical events, as you can be seen. This is the Kaplan-Meier curve of deaths and other clinical events. The curve shows the same the time trend of clinical events. Once again, let me repeat main findings. ALS-FRS was able to evaluate the efficacy of edaravone. Other endpoints, such as the modified knowledge scale and ALS-40, were consistent with ALS-FRS. From the viewpoint of delayed start design, the combination of death and other clinical events might evaluate efficacy effectiveness. Let's move on to the final topic, lessons from clinical trials of edaravone. These are inclusion and exclusion criteria of pivotal study. These criteria were designed based on post hoc analysis of MCI 16 study, the first and failed confirmatory study. These criteria are very strict for enrichment. This is a flowchart of the pivotal study. Although using strict eligibility criteria was used, 2/3 of study patients dropped out. Why so strict? These are the post hoc analysis of the MCI 16 study and the first and failed confirmatory study. The main point, these post hoc analysis is that mild and early, but appropriately diagnosed ALS patients are suitable for evaluation of edaravone. This is a summary table of the eligible criteria in each trial. 19 and 16 study is Japanese ALS severity classification is same, but ALS-FRS score is 19 study, grade 2 or more in all items, but 16 study is no. Years after the onset of ALS, 19 study is within 2 years, but 16 study is within 3 years. And percent FVC is 19 study is more than 80%, but 16 study is 70%, more than 70%. And LS Korea and ALS diagnostic criteria 90 study and 16 study is 96 study is probably ALS is included, but 90 study is excluded. Let's think about potential target population of Darabon. In clinical practice, it's difficult to restrict it to the use of Darabon only for the population evaluated in 1619 study. We need to consider these 4 factors. And based on the results of clinical trials, as you can be seen, clinical trial other than MCI 1619 study provided experience of these populations. A summary of the final section. We need to consider a lot of things. Speed of ALS progression, certainly of the diagnosis of ALS, respiratory functions, disease manifestations of ALS, and the time course of ALS. Taking all these issues into considerations, in the post-marketing survey will be conducted for the efficacy for death, tracheostomy, and mechanical ventilation in the long term using historical control. This is a draft outline of post-marketing survey. Observation period is up to 5 years. Planned number of patients, 700 patients. Major survey items is this. To summarize today's point, the dosage of edaravone is peculiar. ALS R is not so bad, and this and other clinical events are also variable in evaluating efficacy. Enrichment, it's effective but difficult. And the English version review report of edaravone has not been shown yet, but there will be shown soon. Thank you for your attention.
Thank you very much, Dr. Ichimaru. Are there any questions? Dr. Brooks?
Thank you very much for an extensive and really well-developed presentation. As we move forward with these guidelines here, one of the most important things that occurred was you excluded patients who had spinal surgery. And this may be a much more important point than we sometimes think of. Now, our Canadian colleagues are looking at the effect of riluzole on cervical spondylosis recovery and things like that. So I think we have to keep in mind as we move forward the inclusion-exclusion criteria, particularly with respect to spinal surgery. And this, I think, will impact on the issue of sham surgery as well, because if surgery has a negative effect on the course of ALS, we have to be very careful in terms of the recommendations we make.
You understand?
Yeah.
Spinal cord surgery is sometimes alter the progression rate. So how about you in this case? Excluded or not?
Sorry, yeah.
Probably. Yes, please.
I'm Ken Sakushima from PMDA. We understand the effect of surgery of spinal cord. However, it is difficult to include those factors into the inclusion and exclusion criteria in Japan. However, so MCI 19 study some surgery, patient with some surgery received or excluded to conduct clinical trial effectively.
Okay. Go ahead, please.
Oh, I'm sorry. Jonathan Glass, Emory University, Atlanta, Georgia. So it seems like, first of all, thank you very much for presenting these data. It's very important for us to see this. And it seems like you have a negative study and a positive study. And you've chosen to focus on the positive study, but there is this post-marketing survey. I'm curious on how you deal with data from a post-marketing survey. And will that potentially influence the future of this drug? I mean, what if the post-marketing survey is negative? How much data do you really get? Is it as good as another clinical trial?
Can you explain a little bit about the post-marketing study? It's a very important part of this clinical trial.
So we understand that post-marketing study is not a clinical trial. However, in Japan, we have the patient registry data of ALS. Take into these things consideration, we can evaluate the effect to survival in ALS based on that data as using as external control. In addition to that, so safety can be evaluable in post-marketing survey in real-world clinical practice.
So can I just ask a follow-up question? So it looks like this drug has been approved for stroke for more than a decade. And how effective is it in stroke, and how has your post-marketing surveys for stroke informed you of how this drug works?
So as we mentioned, so indication of stroke is only 14 days. However, in ALS, this drug is used in the course of ALS. So we need to repetitive use of this drug in ALS. So maybe is safety evaluation in ALS in advanced patient is important in post-marketing survey. Is this answer to your question?
I'm actually asking more than safety. I'm looking for efficacy. How much efficacy is there in stroke?
Stroke? In post-marketing survey for ALS is not for the collecting the information for stroke.
Yeah, post-marketing survey is a very important part of this clinical trial. So now, as he said, the independent survey team will handle of the both data, natural history data, and this prospective data, and the background will be adjusted to each other, including maybe stroke. So probably it's a kind of study, open study though. Probably it's a continuing discussion. Please.
So mine's more of a comment actually to the European and the Japanese FDA. So if I have my facts correct, and I might be wrong, it appears you approved this based on ALS FRS and would like follow-up. And that's smart of you. It's unfortunate that the Europeans require survival as well, because many of us in the U.S. believe that ALS FRS should be sufficient. And unfortunately, that means the Europeans will fall behind in advancing drugs.
I won't answer to that, but I do have another question. So since one of the first study failed in more severely affected patients, which included more severely affected patients, it failed, and then you had an enrichment with mild to moderate patients. And how do you have any prescription duration in your SMPC? How long should it be given then to patients? How long is it effective? Is there any prescription given taking that into account?
So, as we mentioned, it is difficult question. However, we have experience dosing this drug to advanced patient, the efficacy is not evaluated in those clinical trials. However, we did not restrict the use of this drug, only the patient in MCI 19 study. So we need to collect more information in advanced ALS patient at post-marketing survey. Sorry, it is difficult to answer.
Please, last questions. Yes, James Berry. So this is quick, and this actually is a good setup for it. Is your thinking that there's a biological difference in response between people who are early in the disease and people who are late in the disease? Or is your thinking that the trial, just by restricting your criteria, you are able to see a difference that may be there throughout the course of the disease? I think that's one of the things we're struggling with here.
Based on the characteristics of this drug, neuroprotective drugs, we consider that there are no difference between the early patient and advanced patient regarding the mechanism of efficacy.
One comment. In my impression, this is a very unique design of a clinical trial, but I think that it's a very standard to be as a clinical trial for ALS. The starting point is a failure of a phase 3 clinical trial. That's a very important part. That's it.
And if I could make a comment before, thank you very much. Just a comment to Dr. Berry's query. I think all of us feel in this disease, in the animal model, and in all neurodegenerative diseases, catching it early is the key. The biology, I'm not sure, changes dramatically, but the potential efficacy is going to be far greater earlier rather than later. And I think Dr. Haberkamp's emphasis on prevention is the ultimate goal, but we're nowhere near that in understanding how to prevent. So our next speaker used to come from the great state of Texas that leans left, as most of you know. I wish. Dr. Bryan is a neurologist, clinician, scientist who was in Dallas at Southwestern, and then migrated his talents to the FDA, where he is now really an important member of our group, stationed there, helping us in cell and gene therapy at the present time. Wilson, welcome, and we appreciate your comments.
Thank you. I really appreciate the opportunity to talk with y'all today, and I am here representing the FDA. A little bit about the FDA organization. There are 7 centers in the FDA, and the 3 centers on the left of this slide are the 3 that regulate products for the treatment of humans. So those 3 centers are the Center for Drug Evaluation and Research, and most of you who've had some interaction with the FDA have probably dealt with the Center for Drugs, the Center for Devices, and then I'm here representing the Center for Biologics, and I work particularly in the Office of Cellular Tissue and Gene Therapies, dealing with gene therapies, cell therapies, embryonic stem cells, fetal-derived stem cells, induced pluripotent stem cells, devices, and sometimes therapeutic vaccines. It's very cool stuff, and it's a lot of fun. But I want to talk today, of course, about ALS and the spring training. For those of you who don't know, Major League Baseball spring training starts tomorrow and this weekend for some teams. And that reminds us of Lou Gehrig, who was a hero in America. And he was a hero partially because he was known as the Iron Horse, that he played in over 2,000 consecutive baseball games. And when thinking about that, I thought about the many years that many of you have put into ALS research. Some of you have been doing this for 20, 30, 40 years or more. And what I'm going to say today is that in spite of, I think, the frustration that we all have with the lack of better therapies, I really think that this ALS research community is doing things right, and I'll endorse in general what's been going on. So let's talk a little bit about the drug development process, which should be an orderly process, starting with some good understanding of the disease pathophysiology. And based on that understanding, we can hopefully identify pathways that we might target for intervention. That target identification is critical. Then a pharmaceutical company will go through a process of drug discovery where they'll look at not just one molecule, but many hundreds or thousands of different variations of molecules. Take the best ones, the ones that look best in vitro studies, and take those into animal studies. And those animal studies we look at for evidence of safety and also for proof of concept, and ultimately to guide the design of the clinical studies. Now what goes wrong? Well, the main thing that goes wrong, and I think most of us know that the main reason that the trials fail in ALS is we don't understand this disease well enough. And if you don't understand the disease, then your target identification is flawed, and then you get maybe some equivocal results in phase 1 and phase 2, and you go into phase 3 and it fails, and you start to blame your phase 1 and phase 2 trials when actually the problem was we don't understand the disease well enough. Another problem that we see sometimes is that the animal studies that are done are poorly designed, they're seldom reproduced, and shall we say, imaginatively interpreted. Preclinical studies, the same rigor in preclinical studies as is needed in human studies. These should be randomized, they should be a concurrent control. In general, I think these should be blinded. The design of the preclinical studies should look like the design of the clinical studies that are going to be done, the same route, regimen, and if there's a device involved, if it's feasible to use the same device. And ultimately, these animal studies should be reproducible if you're going to take a product into the clinic. Clinical development, again, this should be a systematic, rational process where phase 1 studies get evidence of safety, tolerability, maximum tolerated dose. We encourage a concurrent control and randomization even in phase 1 to get some preliminary evidence of efficacy, taking that data to guide the design of the phase 2 studies, which look closer at endpoints, population, and then ultimately the phase 2 data guides the design of the phase 3 studies. Now, this is sort of a step-by-step how you go from phase 1 to phase 2 to phase 3. But besides these objectives of just moving a drug forward, there's sort of other objectives of advancing the field. And I know that many folks in this room and this workshop will think about this, that these trials are very useful for evaluating biomarkers and for developing endpoints. And I think we're all looking for that ideal endpoint that's relevant to the disease, that's reliable, has inter-rater and intra-rater reliability, that's resistant to bias, it's sensitive throughout the course of the disease, it's clinically meaningful, it's feasible, meaning it's not too expensive and patients will tolerate the procedure, the outcome measure, and also that it's clinically translatable. And by that I mean that we'd like to have endpoints that if we use them in a phase 3 trial and they show the primary evidence of efficacy, at the end of the day, the doctor has to sit down with the patient and explain what the benefit of this drug is. And if our endpoints are so complex and convoluted that you can't explain to a patient exactly what the benefit is, then we sort of disenfranchise or disempower our patients. And so I encourage you to use some endpoints, not all of them, but some that we can explain to patients. Now, there is no ideal endpoint. There is no single endpoint that has all of those characteristics. And so when folks sometimes talk about an endpoint being validated, I cringe at that word because I'm not sure what it means. The endpoint that you're going to use as the primary endpoint for a phase 1 trial is very different than the primary endpoint for a phase 3 trial. And because no endpoint is perfect, you're going to look for other secondary endpoints that complement that primary endpoint. So as new endpoints, new outcome measures come along and are being developed, don't get too invested in any one endpoint. Characterize the endpoint. What are its advantages and disadvantages? And in any particular trial, think about the objectives and think about which endpoint is best for this particular trial as the primary endpoint, and then which secondary endpoints have features that will complement that primary endpoint. So what goes wrong in clinical development? Sometimes we don't explore the dose and the regimen enough. Over-interpreting biomarkers as surrogates. Biomarkers mislead us more often than they help us when we use them as surrogates. Proceeding to phase 3 when phase 1 and phase 2 results suggest that the product is ineffective. And this happens, at the FDA we see this all the time. Sponsors come in with these phase 1 and phase 2 data, and we look at the data and the sponsor tells us how wonderful this product is, and we don't see anything. And I think people get too invested, just as they get too invested in a single endpoint, they get too invested in a single drug. Try to be objective. A few considerations for ALS clinical trials, and I'm not going to go into these in detail, but we encourage innovative trial designs, futility analyses, adaptive trial designs. You don't have to go from phase 1, phase 2, phase 3. You can have a phase 1-2 trial. You can have a phase 2-3 trial. Enrichment designs, those were talked about by the previous speakers. We encourage enrichment studies. We have to get better at clinical trial methodology in order to make these trials smaller and more feasible. So, since I'm from the FDA, I have to talk a little bit about the regulations. And this is from the Code of Regulations, or the CFR. And the FDA, for marketing approval of a product, requires substantial evidence based on adequate and well-controlled studies. Now, what constitutes an adequate and well-controlled study? Well, it should have a pre-specified objective, pre-specified analysis plan. You can't do the study, get the results, and then decide what the appropriate analysis plan is. There should be a control, and there should be methods to minimize bias, randomization, blinding if it's feasible. The FDA, for marketing approval, generally requires 2 adequate and well-controlled studies. But folks are always asking us, well, how can I get away with just one study? When would one study be sufficient? And so this slide describes some of the considerations on when a single study would be sufficient. First of all, it's got to be an adequate and well-controlled study as described in the previous slide. But we also need for the results to be robust. And what do we consider when thinking about whether the results are robust? That is it a large multi-center study? If all the effect is coming from a single center, then we're more skeptical. Is there consistency in the results across subsets? Is the activity present in both men and women? Is it present in both older folks and younger folks? Is there consistency across subsets, consistency across study centers as well? Is there consistent result across endpoints? So we'd like to say if you show an effect on survival, or if you show an effect on the ALS FRS, be nice to also see an effect on the FVC or some strength measurement or on survival. Consistency across endpoints counts. Statistically very persuasive results. P-values of 0.043, that's statistically positive, okay, but you'll find when you get p-values that are close to 0.05, just a small number of subjects can change that p-value dramatically. And so we like for it to be statistically robust. In general, we will accept a single study in situations in which a second trial would be ethically or practically impossible to do. So I want to make a few recommendations. Keep developing biomarkers and endpoints. And I know you folks are doing this because I looked at the neurology, the March 1st issue of Neurology, and I was heartened to see the report by Dr. Bedlack and colleagues in Boston and Durham looking at the ALS FRS and what I'll call the natural history of ALS as marked by that. And I appreciate also the commentary from the colleagues in the Netherlands on that paper. But I think that sort of research is critical to improving the design of clinical trials and hopefully getting to enriched clinical trials. And I was also heartened to see the article that came right after that by Dr. Murphy and her many colleagues from around the country, it looks like, looking at cognitive function in ALS. The better we understand the disease, the better our trial designs will be. FDA encourages innovative trial designs, adaptive designs, enrichment designs, futility designs. Plan ahead. I didn't talk about it, but if you're doing animal studies with the product, then you ought to be thinking about what is my phase 1 study going to look like? What is my phase 2 and phase 3 study going to look like? Plan ahead. We, like the EMA, we encourage early and regular communications with the FDA. And again, scientific rigor throughout the preclinical studies and the human studies, I think, is essential. And I think that this group has stood up for that, and I hope that you'll continue to do that in your deliberations over the next few days. And I think this is all I'm sorry. There's one more thing. So Ben Brooks mentioned that we had a public hearing about 3 years ago. And Ben and Dr. Appel and several others were there. And one of the things we heard from, at least one of the things I heard from patients, was the frustration that they have with the current therapies that are available and the small magnitude of the benefit that's out there. And sometimes when folks talk about developing products in ALS, they go to this conversation about incremental change. And I got to tell you, when I hear that word incremental, I just cringe. To me, it's saying that we're going for some small amount of change that may or may not matter much to patients. And I kind of think in baseball, it's kind of like a batter going up to the plate and thinking, well, I hope I'll get a walk here, and maybe the next guy will get a hit. And that's okay. We can do those studies. But what made Lou Gehrig a hero, part of it, besides being the Iron Horse, was that he was a home run hitter. And he struck out a lot, but he was trying to swing for the fences. And what I enjoy in cell and gene therapies is that I see a lot of investigators in a lot of diseases who are trying to stop diseases and trying to cure diseases. And I know that some of you are trying to do that. And I think that's what heroes do, and I thank you for it. And I appreciate it. I do want to mention a couple of our guidances. We have guidances on adaptive designs and enrichment strategies for those that want to learn more about those things. And I'll just skip down to my contact information and stop there. We have a question in the back of the room.
Yes, William, thank you. My name is Ed Tessero. I'm an ALS patient of Dr. Glass's in Atlanta, Georgia. So I want to remind you that the White Sox in Chicago won a series by going up and getting a walk and waiting for a hit. So, you know, that when you don't have a lot of power hitters, you take what you have, and they won the World Series. But I want to ask you a little bit about the specificity about the endpoints and how important that is to you. We heard this morning about the heterogeneity of this disease and how it presents so many ways it frustrates the hell out of you. I hear that loud and clear. But are we overly requiring of these endpoints? In other words, my version of ALS, and I'm in my 8th year, you know, it could be something very peculiar to me that it might not present in another person exactly the same way, and my results in a clinical trial might be very different than 5 or 6 of my counterparts. So within your exactitude that you want to see in every trial, how can you accommodate a disease like this one or understand it with so many different ways it presents? How am I ever going to fit into that criteria or the 30 counterparts of mine that are in the neural stem 566 trial? I'm just curious about that.
Okay, so you've touched on so many different issues. I'm going to comment on a few of them.
Start with the White Sox.
Congratulations to the White Sox. The heterogeneity is a huge problem, and it gets to, I think, part of the basic issue of us not understanding this disease well enough. And I remember 20 years ago we had these discussions of, is ALS one disease or is it a bunch of different diseases? And if it's a bunch of different diseases that we're lumping together here, what do we really have a chance of finding a treatment that's for a bunch of different diseases? And I think we do have a chance, but it's a struggle. And in phase 1, in early development, I think it is very important to look at a wide range of endpoints, because you can't expect that any particular intervention is going to work on every aspect of the disease, and it's going to work in every population. So that you do drug development, you look at those phase 1 and phase 2 studies to see which patients seem to be responding best to this intervention. And that may be patients like you, but it may be that this particular agent works in a different group of patients. But we have to do the clinical trials to sort that out. And we can't know it in advance of doing the trials.
Wilson, thank you. Thank you for clarity, and I think that's very important. There are different interests that go into the making of these trials. And of course, the ultimate purpose of being here is to find a cure or an arrest of disease. And then we start talking about slopes. And those slopes mainly benefit the pharmaceutical companies, I'm afraid. And so I think that's where the division of interest then starts to come into play. And that's very important to sort of focus on, that as physicians and as patients, we have one objective: to take care of the patient and to make their lives better. But there is some amount of magical thinking that sometimes come into play, that we wish we could cure this disease with a knowledge gap that we don't have, and with the targets that we don't have, and then to play with the statistics to approve a drug and then make it available worldwide and sort of go in a track that's a dead end. So I think we have to put on the hat as physician scientists and make certain that our trials are rigorous and that they are not influenced by either public opinion or by corporate interest.
Thank you. I endorse that position.
Go ahead, next question.
Dominic Fee with the Medical College of Wisconsin. You had mentioned about...
I'm sorry, I'm having a little trouble hearing you.
You had mentioned about target-enriched studies, and I completely agree with that. But what about the situation where the studies become, the inclusion criteria becomes very strict to the sense that very few patients actually qualify? And one of the bigger ones is with forced vital capacity either presenting at greater than 80% predicted or greater than 70% predicted to get enrolled. Two parts to this. One part is, do you think we may be restricting the trials to a very select subset of population by having that strict a criteria? And then the other question is, how do you think we should try to involve patients who wish to participate in clinical trials, but their FVC is 60% at presentation?
Okay, so my impression of ALS clinical trials is that it's hard to get the funding to do these trials. That the pharmaceutical industry has a limited interest in these trials, and we need to get more support. But while we're working on shoestring budgets, then probably we need to try to find ways to make these trials more efficient, or they're just not going to get done at all. Now one problem, and that to me means doing enrichment. Now, one problem with these enrichment trials is that if you limit the trial to people who've only had ALS for some short period of time and who only have an FVC within this range and meet a bunch of other criteria, and then you show an effect, what does that mean for the rest of the ALS population? Does the drug work in them or not? And we would, I think, be happy to be asking that question. That is, we'd be happy to have a drug that worked in a very selected population, and that generalizability would be a question, but we could find a way to work that out. Now, it is so frustrating that for patients that not everyone can be in a clinical trial, that some patients you look at and refer again to the manuscript by Dr. Bedlack and colleagues, that some patients just aren't going to be informative in a clinical trial, and the purpose of research is to advance knowledge that hopefully will be of benefit for everyone, and the practical issue is we're not going to have clinical trials that are open to everybody. There are expanded access and compassionate use mechanisms to make products available if the pharmaceutical industry is willing to do that.
Thank you.
Dr. Glass?
So first, I'd like to thank you for, I think, an excellent presentation and your focus on the rigor of the scientific method in making sure that we find the right answers. I'd like to, though, ask you a question about the preclinical work, and you talking about animal models. We don't have any great animal models, and many of us have suggested maybe with a good hypothesis and good scientific background, maybe it doesn't have to work in the animal, as long as it doesn't kill the animal or hurt the animal. So can you speak more about your interest in animal models and preclinical models?
Oh, thank you very much. I'm sorry. I meant to correct that because I talked to Dr. Rustin earlier. We strongly endorse alternatives to animal models for providing proof of concept data to support going into humans. That is, I think that just we as a species expect too much of our other species to take on the burdens of our diseases, and I think that that we're moving steadily toward getting in vitro studies, cellular-based studies, other things that may be more informative than animal models, and we encourage alternatives to animals in preclinical development, particularly for proof of concept studies. So thank you for bringing that up.
Eva, do you have a quick question?
I do. It's the one I asked before. So with your agency, we've completed a phase 1 and a phase 2 trial, Dr. Glass, myself, and our colleagues at MGH, and we've entered 30 subjects, intraspinal stem cell transplantation. So my question, and there was no placebo or sham surgery group in the phase 2. So my question to you is, with the plan on doing both cervical and lumbar transplantation in ALS patients, as the next study, do you feel there needs to be a sham surgery group as a placebo?
Okay, first of all, I can't comment on any specific product and development program.
Right, so it's a general question. Sham surgery in cellular therapies for a lethal disease.
So let me say that the way the FDA works is that we ask the folks who are doing the studies to make a proposal to us and to justify that proposal. And so we're very open to whatever proposal a sponsor puts on the table. Now, the question gets to be, at the end of the day, is that study going to be persuasive? And the more rigorous the design of the study, the more rigorous the conduct of the study, that it means not much missing data, the more rigorous the design, the more rigorous the conduct, and the more robust the results are, the more persuasive it will be. If the design is less rigorous, if the results are not quite so robust, then we get into trials that are difficult to interpret. So some of it comes down to how much risk does the sponsor of the study want to take, that their study at the end of the day is not going to persuade us and is not going to persuade the ALS community. But again, that depends not only on the design, it depends on the conduct, and it depends on the results. If you've got this huge effect in ALS, I mean, if you've got a product that stops this disease, then the trial design is going to be easy, and it won't matter a whole lot. It's these incremental effects that are so hard to detect, and where we get into trouble when we compromise on the trial design and the trial conduct.
So, Wilson, thank you very much. We've set the bar really high for scientific vigor, and I think the next 2 days will allow us, hopefully, to make real progress in accomplishing that. So thank you. Hiroshi.
Thank you very much, Stan, again. So we have 10 minutes. And so you go and the physiological need, and if you can get the coffee, if you're lucky, get back to 10:20. All right, thank you.
Criteria be restricted, and will be sticking to the 8 minutes
Okay.
in the Hiroshi paradigm.
So I will talk about the eligibility, and actually Giancarlo Logroshino was unable to come, so I'm taking care about question number 1. Related to question number 1, there are 4 issues that we analyzed. First of all, the new diagnosed patients, and we have a problem of incident versus prevalent cases, old plus news in the case of prevalent. Prevalent cases are more likely to be present in a multidisciplinary clinic site of recruitment for clinical trial than in a population-based setting, and the incident cases have a worse prognosis compared to prevalent cases. If this is the case, the difference of setting is probably associated with a difference in survivorship of about 1 year, if there is evidence for this. So the rationale is that the inclusion of excessive number of prevalent cases could account for the decreased power in clinical trial. And the recommendation number 1 of our group is that in the clinical trial, the goal should be to shorten the time interval from disease onset to trial entry from one side, but the other side we have to balance the request of patient to be included in clinical trial according to these 2 problems, incident versus prevalent. This is point number 2, familiar cases. The percentage of familiar cases is around 5 to 10 to 15% of all our cases, and they have probably the same presentation of sporadic cases. There is no evidence so far indicating a specific prognostic role for the presence of familiar history of ALS. On the other side, it's quite clear that there is evidence that specific gene abnormality will deteriorate the clinical phenotype of ALS at onset and will change the prognosis. And several studies tend to indicate the CNIORF can affect prognosis. So if this is the case, the rationale is the subject with positive family history may have a different prognosis because of the underlying genotype of early environmental exposure in life. Actually, this is provocative. We don't have evidence about this point. And recommendation number 2 is that there is evolving evidence to consider specific genotype as very important for certification when planning a clinical trial. There is no enough evidence that family history per se justifies certification. As a logical consequence, economic research should be planned accordingly. The point number 3 is related to cognitive impairment. The percentages of the ALS patients with cognitive impairment are abundant. Around 50% of our patients can have cognitive impairment, and those cognitive impairment can affect specifically the disease course. Specific cognitive deficit at baseline predict a more rapid disease course. This is proven. There is evidence also that tend to suggest that those individuals without neuropsychological deficits at baseline are likely to remain free of deficit through the course of the disease, but this needs to be further proven. The rationale number 3 is that inclusion of subject with cognitive impairment at baseline obviously has to be considered because they have a worse prognosis that can affect the balance and the results of a clinical trial. And the recommendation is that neurocognitive assessment plus behavioral impairment, but there is no evidence enough related to that, are particularly important. They are prognostic factor that should be used to stratify ALS patients at entry in clinical trials. The last issue are the restricted syndrome. ALS is generally considered a disease with focal onset and spread to progressive regional involvement. And the mix of clinical signs of the first and second motor neuron are representative for the diagnosis. But at the same time, both signs are not usually present at the beginning of the disease. This may determine diagnostic delay in many patients. The clinical involvement in some of our patients can be limited to upper or lower motor neuron, and subjects that can be classified according to Velez-Correa criteria as a clinical defined have a faster curve of disease. On the other side, the presence of pure upper or lower motor neuron signs are generally associated with longer survivorship. So the rationale number 4 is that restricted syndrome may have a better prognosis compared to typical ALS, and the recommendation is that specific clinical phenotypes should be included in different strata at entry in clinical trials. Finally, after discussion last night, we had to add a further problem related to, obviously is emerging, I was emerging before, we have an issue of predictive testing, eventually in subgrouping the patients, but at the same time we have the issue of responsive predictive markers. In other words, during ALS drug development, we may include a relatively broad spectrum of patients with early stages, and then we need to narrow to a more target class of patients defined by new discovered responsive predictive markers. So we are in a kind of ambivalence to have all the patients in our trial previously selected, but at the same time, after the trial conducted, we have a problem to identify responsive population with predictive markers. Thank you.
Thank you. The floor is now open for discussion. Please make sure you state your name in case we're video streaming.
Hello, Rick Bedlack again from Duke. So with respect to your recommendation number 1, it makes sense to limit inclusion to prevalent cases for trials that look at survival as the primary outcome, like our phase 3 trials. But in the interest of trying to maximize inclusion of all patients, wouldn't it be okay to get rid of that for like early phase trials where we're looking for safety, tolerability, maybe biomarkers?
You talk about in terms of incident versus prevalent, specifically? Yes, probably you are right. The point is that definitely we have to be very careful about the prevalent cases. This was the point that we were trying to make in our group. And obviously, if we put survival as an endpoint, that's critical. If we use other endpoint, it's less critical, as you said.
So Gary Gross at the University of Kansas. I'm the methodologist. So just a couple of formatting issues. So first, all the rationales are incomplete. All you have is an inference statement. An inference only makes sense if there are other premises to infer from. So some of your background statements are actually supporting the recommendation. Some of those should be moved in. Some of them are evidence statements. Some of them are principles. But an inference just can't stand by itself. You need to have other statements. Secondly, none of the recommendations are actionable. They're in the passive voice. Who's supposed to do what? So I would just, reword those. Those are just formatting issues that I'll point out.
Yeah, this is a problem of phrasing, obviously,
sure.
And put everything together. Actually, the background has a lot of evidence, but we were unable to put it in. But this is what we have to do probably in the next few hours.
Just move some of those statements to support that inference.
Sure.
And then make the recommendations actionable.
Thank you, Gary, for giving us a good kickoff idea about what needs to constitute a recommendation, which has to be an action statement. A question that I have for you, Vincenzo, relates to the difference between stratification and exclusion. So the question was about eligibility criteria, and, for example, on cognitive impairment. You talk about stratifying for cognitive impairment. My question would be, should we exclude patients rather than stratify for them unless the focus of the trial is on cognitive function improvement. So that's a question for you.
I don't personally think we need to exclude those actually patients, but at least we need to stratify according to the specific evidence. Obviously, we need to think to ourselves what is the target of our trial, and that can probably change our specific issue. I don't know what is Kathy's opinion about this. This is relevant.
So stratification will help us in our analysis, but it brings in a subgroup where power is lost, and that is a concern. We already have issues about power because of the heterogeneity of the disease. So we don't have to settle this now, but I want to just have us think about whether these eligibility questions really can be totally handled by stratification, or whether we need to ask ourselves about excluding certain types of patients in order to improve them on the heterogeneity challenge, which is really huge for us.
That's a major issue. I'd like to reinforce the fact that we have evidence of cognitive changes, but not yet for behavioral changes as well, because this is critical as well. Cognitive means no behavioral whatsoever. So I don't know, I think if this is a relevant issue that we have to analyze, further analyze.
I think we have to realize that in developing guidelines, we're developing a license on a certain way of doing things, but it can also be a leash, and we have to be very careful. I think your language, a la Gary Gontrez, should be stratified. And then in the individual study, you may say stratification includes exclusion, and I think our language should go in that direction, that the actual ultimate trial may be dependent on resources, but it definitely has to be stratified according to these principles for which we have good evidence. And then the ramification of that stratification may include exclusion in some trials, but we should not exclude. I think we should just stratify.
This is Eric Macklin. Same issue, maybe about semantics, about stratification, whether it's exclusion. The third option is that we adjust for some predicted outcome on the basis of a range of predictors, of which the number of those that you've identified would be relevant. I don't think stratification in a formal sense of dividing a sample into different bins at randomization and analysis is likely to be successful, because, as you've identified, we're going to end up with a single individual in each stratum once we divide by site. If we have a 20-site study with 10 people at most being enrolled at any site, and we now divide them up into 10 different bins, we're not going to have an analysis to pursue. So anyhow, as we operationalize this, I think we need to be a little more careful about our recommendation with respect to what stratification might mean.
Yes, Ryuji Kaji from Japan, and I agree with the review that we do have to select out some patients. And there has been discussion about enrichment, and because survival is to see the kind of disease-modifying agent, and then, for example, in the field of Alzheimer's disease, you have to give them a drug very early in the course. That's been shown in many other cases. So as in AD, in ALS, we have to select out early stage patients. And so that's the key for the enrollment. I agree, just comment.
I think this is the future, because it has been discussed this morning. I think we have to be preclinical somehow. We have to at least Alzheimer's demonstrate that we have to treat our patient with the biology starting, but we don't know exactly when the biology is starting. We don't have a biomarker.
Yeah, what we need is a good biomarker or good early diagnosis criteria.
Sure.
So Gary Gronseth, maybe I'm stating the obvious, but the only benefit of stratification is to ensure that the patients are balanced on confounding characteristics to treat, and that's it. It doesn't do anything for the assay sensitivity beyond that. So
that's critical, probably.
David Watt. So just to
reinforce the point Rick made with regard to stratification and exclusion. It may be that this is less of an issue when you're looking at safety, tolerability, target engagement, and it may be that you might be in a situation of excluding certain subgroups when you're looking at efficacy in with limited resources.
Well, you're mentioning eventually exclusion for other reasons, like metabolic exclusions or something like that?
No, I just mean if you have a concern about subtypes, exclusion, stratification might end up meaning exclusion if you've got limited resources and you're looking for a signal of benefit. But if you're looking at things like target engagement, safety, and tolerability, it might be more acceptable to include a broader, more diverse or heterogeneous group of individuals.
Thank you. I think we're going to have to move on.
Just operational issues. Anyone who belongs to Vincenzo's group, please write comments and so forth, because tomorrow's breakout sessions, you have to incorporate those comments into this discussion. So please help him, because he's answering a question. He cannot write it down, so please.
Dr. Hardiman is going to present research question 2, which is which diagnostic criteria should be recommended for inclusion.
So it leads on quite nicely from the discussions. And the background is, of course, what we're saying, that ALS is most likely a heterogeneous group of conditions, probably with different pathogenic mechanisms. And I think the discussion that we had a few minutes ago about stratification should also be about inclusion. And some of the heterogeneity may well be categorical, one being C9orf72, another probably being the presence of extramotor involvement, particularly cognitive and/or behavioral impairment. So the background is that this is a heterogeneous disease. It's heterogeneous in genetics, possibly in environmental exposures, although we don't have really good evidence to support this at the moment. There is a large European study that's data mining at the moment on this. It's also heterogeneous in clinical presentation with respect to site of onset, bulbar or spinal, and also with regard to the presence of extramotor involvement, particularly cognitive and behavioral status. And we know that cognitive status, particularly executive dysfunction, is a negative prognostic indicator, and that may well be categorical. And also in the rate of disease progression, which is informed by some of these things. So the rationale is that the Ellis-Gorel criteria is somewhat limited in recognizing particularly the presence or absence of extramotor involvement. And this came up earlier this morning as well. So we should consider updating the criteria. This is underway currently by the World Federation of Neurology Research Group, and the paper by Federica Augusta and Albert Ludolf, these were first authors of 2 consensus documents that we published in the ALS journal recently, should be noted. So the recommendation in this regard is that these criteria should really reflect the recent advances, particularly regarding genetics, which is categorical C9orf or not C9orf, and possibly neuropsychological impairment, executive impairment or no executive impairment. That seems to be categorical, at least on the available evidence. The second background is that heterogeneity must be taken into account in future clinical trials, and I think this refers to the question of inclusion versus stratification, and there are arguments in both directions. The power calculations that we make incorporate the noise that heterogeneity generates, and one of the possibilities that we could consider is we could reduce the noise by reducing heterogeneity, and that would therefore have less of an effect on power calculations. So I think if we think about heterogeneity and how we limit heterogeneity, we can undertake a recalculation of our power analyses. So so that's the background. The rationale is clinical trial eligibility is based currently on the Ellis-Gorel Earlyhouse Owaji criteria. The recent inclusion of possible ALS hasn't led to any loss of specificity. The certainty of diagnosis at entry into a therapeutic trial should be defined as all of the incorporated aspects of the Earlyhouse Ellis-Gorel criteria pending the introduction of new clinical trial guidelines. The third background is the issue about genotype, and I don't think anybody will argue with the presence of a categorical difference with respect to phenotype. Clinical trials are currently underway based on genotype. For example, the ASO for SOD1 and the development work by ICES and Biogen for C9orf72. If we genetically test patients in advance of clinical trials, which we will be doing for these studies, that would provide a selected and very, very selected cohort. So that would be a selectivity based on inclusion rather than stratification during the trial. It may well be that looking at other neurologic diseases, for example, epilepsy, pharmacogenomic studies could allow us to identify different responsiveness to therapeutics. So we should really consider incorporating that in some manner in the formulation of our trials going forward. So the recommendation should be that genetic screening of patients should take place prior to enrollment, and this may drive a clinical trial such as a gene-based therapy, but even trials that which they are not gene-based, we should really screen for C9orf to commence with anyway. Or we should collect and store blood for post-hoc genetic analysis. This would be the case for example, investigator-led studies where cost issues may prevail and it may be difficult to screen in advance. The fourth background is the changing age profile of ALS, and those of us who have population-based registers will attest to this, that the peak age of onset is moving somewhat to the right. There are reasons, biases and other reasons why this might also be, but it may also be that the genuine chronological and biological aging process have changed somewhat. The life expectancy in general of Western societies are increasing, and the age of onset, as I said, may be increasing. And there is a case to be made that very old, who are otherwise well and chronologically old, but biologically perhaps in good shape, are currently being excluded from trials. And so recommendation is that those who are considered old or very old, for example, those over 85, but otherwise in good health, should not be categorically or by definition excluded from trials. The fifth background is this, again, the concept of heterogeneity with respect to disease progression, rather than heterogeneity with regard to inclusion into trials, or heterogeneity with regard to pathogenic processes. So the issue about getting patients into clinical trials early, but allowing for variability within the disease progression, which requires larger numbers because of disease heterogeneity. So the current trial design can delay the initiation of trial drug because we sometimes need to do a 6-month run-in period to establish the rate of progression of the disease. But there are new prognostic indicators and staging methodologies that have been developed that could cater for this. And so the recommendation is that we should either stratify or enroll, probably stratify according to predefined prognostic groups, and that we could incorporate these validated staging protocols and other prognostic indicators such as executive function. And these could be used to facilitate inclusion into trials at earlier stages, such that patients enter trials in the early stage of the biological process of disease rather than the later stage. So I think that's all right. Anyway,
So Gary, just some formatting comments from Gary Bruijn. So first, label all the rational statements in terms of what they are: principles, inference, evidence. All the recommendations are in the passive voice. Who should do the revision, for example, because that helps with the impact. And then backgrounds 2, 3, and 5 are actually recommendations. And so you might want to consider moving them around, or but that's all I have.
Jeff?
That was great. Small question. Under 3, genetic screening, you didn't actually list the genetic screen, which genetic screen. You mentioned C9. Would you recommend C9 or all of the available ones, which is obviously expensive?
Yeah, so this was discussed within the group, and so it was somewhat vague for that reason, because the cost of doing an entire screen is probably prohibitive, particularly in investigator-led studies. the evidence would suggest that we should screen for C9orf72. Okay. And of course, ALS may well be oligogenic, and it's also probably even sporadic ALS has a complex genetic basis to it. So the point about the pharmacogenomic analysis would cover that. I think going forward, we probably should store DNA for everybody who enters into a trial, and I think we should have that DNA as a resource for subsequent pharmacogenomic analysis. And if you look at, for example, the epilepsy field, that's what's happening. There's a pharmacogenomic program in epilepsy now, which is allowing targeted treatments.
Not to keep this too long, what about like risk, like ataxin-2, a risk factor, but not a causative gene?
Well, I mean, I think that's a difficult one. We know that there are some single genes of major effects that are Mendelian inheritance. We also know that even within those cohorts of patients, for example, there's a higher likelihood of C9orf to travel with other so-called susceptibility genes. So I'm not sure that we can be categorical about causative versus enabling genes, because I think we just don't know enough about that. So I think we should be agnostic to some extent about that. We know that there are single genes of major effect that probably do cause the disease, SOD1, A4V, C9orf72 most of the time, although it can also cause FTD and other neuropsychiatric phenotypes. But at least we have some categorical evidence to that effect.
Chris Coffey, University of Iowa. I guess this is more of a comment for the group's consideration than a question, but one of the topics that's already come up, and I think will likely come up in other areas, is this idea of enrichment designs. And it seems like a lot of what you're talking about, although it wasn't mentioned in your document, has to do with how do you decide what to enrich on as well, as much as the inclusion criteria. So I thought that might be something to consider, at least bringing into somehow into your discussion to tie it with the other areas.
Yeah. I think the discussion that we've had in our group is exactly around that. It's the fact that the disease is heterogeneous, and in order for us to reduce the degree of heterogeneity, the likelihood is, like I said, that we could probably improve our power calculations with smaller numbers. Of course, the key is what are those factors that are driving the heterogeneity, both at a biological level, and also what are the clinical or paraclinical markers of that heterogeneity. So we know C9orf72 is one of those from a genetic point of view, and there may be 15 other single genes of major effect. We do have some markers of categorical difference, being extramotor involvement being one of them. Executive dysfunction is probably categorical. And there may be others as well, that if we deep phenotype our cohorts well enough, and those of us with registers are doing that, I think the likelihood is that we will be able to perform subsequent cluster analyses, which may actually identify patients that would otherwise appear to be similar as a clinician sitting across the desk to us, but may sit into other clustered bins that have a similar either disease pathogenic mechanism or disease progression. So I think we should start thinking about that.
Martin Turner from the UK. Just a point really for us to think about as a group, that the routine genetic screening of sporadic patients, which I think probably will have to happen, will be a massive change in our practice. It will bring in all sorts of issues about disclosure, patients wanting to participate in trials without being told their own status, and that's something that we'll have to think of as a group.
My personal view on this, and I think I probably reflect the view of our group, is, if we want to undertake trials and patients engage in trials, and we want to stratify appropriately, these are things that these are difficult ideas and concepts that we're going to have to grapple with. There is clearly a categorical difference between C9orf positive and C9orf negative. I think that storing DNA for all of the other conditions is a service to the ALS community, and I think we need to really embrace that or engage with that. Difficult and all though it may be. I'm not sure that the difference between sporadic and familial is as clear cut, actually.
This is just a comment with respect to the verbiage, since we're an international group. In principle, I think the issue with respect to genetic testing might be a function of what the gene expression is in the local population, and we should get that kind of verbiage into the recommendation.
Yeah, I think that's a really good point, and that's another function of heterogeneity. That population-specific heterogeneity is going to be important as well. That's a really good point.
Yeah, Jeffrey Rosenfeld from Loma Linda School of Medicine.
Orla, that was an excellent summary. And I think of all of the topics we're going to address in the next 2 days, the issue of heterogeneity and how to handle it is probably among the most important. And along that line, especially for recommendation 5 and one of the earlier recommendations, I would encourage you to be more prescriptive with regard to how to stratify, because assuming that this guideline will be used by folks that don't take care of patients and maybe don't have the same appreciation of the heterogeneity, maybe not just on prognostic groups, perhaps on phenotypic characteristics, as we don't really understand what defines different diseases or subtypes of the disease. Maybe the recommendation could even be more direct as to make suggestions for different categorical groups.
I think the issue is going to be the one that arose in Vincenzo's discussion as well. It's a question of whether we should be selectively recruiting or whether we should be selectively stratifying. Stratifying is expensive. Selectively recruiting would probably be cheaper. And I think that's something we'll have to grapple with as well. You know, if we are going to do that with C9orf, with the SOD1 mutations and the C9orf mutations, we're going to selectively recruit, and that's okay. And one of the things we might want to consider selectively recruiting would be around other clinical markers that we know about. For example, the presence or absence of cognitive of executive impairment. We could selectively recruit either for or not for, or even selectively recruit as long as we know incident versus prevalent patients, they have different characteristics. It doesn't matter how we do it as long as that's what we decide we're going to do.
But selective recruitment would seem to make sense only if you thought that the drug was targeted against whatever was driving that particular selective recruitment.
I completely agree.
I wish we were there, but right now we're still fishing. So fishing with a net is better than with a hook if you are not sure what you're going to catch.
Yeah, but then we have to stratify, and that makes the trials very expensive. Yeah.
Final comment.
Okay. Concerning with heterogeneity, I agree completely to screen a single gene, as you mentioned. But how about the modifier gene, something like it may be a rare variant or some other things may modify the clinical course or the prognosis, something like that. And in case of cancer, there are so many modifier genes, and responsiveness against a certain drug is quite different or governed by these kind of modifier genes. It may be a future issues, but how about that one?
Yeah, I completely agree, which is one of the reasons for the recommendation that we collect DNA on every patient who enters a trial and have a DNA repository. The issue about modifier genes and genes that may contribute or otherwise to both the phenotype or the risk phenotype or progression, I think there's quite a lot of noise in those data sets as well, and I think we need to have many of these so-called modifier genes validated in large, so there's some experimental work to do as well in larger cohorts, and these may also be population specific, so and there may be gene-gene interactions. Project Mine, I think, will help with those sorts of analyses as well going forward.
Ettore Beghi from Milano. I wonder whether we should address this issue in subsequent steps. The first objective that we have is to prove that the drug is effective. In that case, we should reduce as much as possible the heterogeneity in order to see if in the two treatment arms there is a difference. Then we can go to the other patients, and so my suggestion would be that in a phase 2 trial we should be very selective with very homogeneous patients, and having proved that the drug works, we may go on and eventually in phase 3 studies have a more broad population, stratifying the patients according to major prognostic factors.
Yeah, I completely agree. I mean, I think if we use the analogy of pneumonia prior to our understanding of microbial infections, some antibiotics work on some types of pneumonias and not on others. And if we did a trial just for pneumonia with an antibiotic, we'd have either a very large N and a lot of noise, or we'd have a negative trial. So I think this is a really crucial point at where we are in this disease. And we really have to work very hard. And those of us who are clinicians, I think we have a lot to contribute in this regard to allow us to try and identify what are the real drivers of heterogeneity and the development of biomarkers that segregate out. I think we have a lot of big data now that will allow us to to do these very complicated, probably very complicated large data analysis and the sort of work that King's did originally in the sort of cluster analysis based on the data sets. But I think we can do a much better job with a much richer data set now. And I think that's where we should start, actually.
May I make one last brief comment, which is that there's been very little discussion yet this morning about identifying the early patient. We talked about prevalent versus incident, but there is a lot of evidence suggesting that early patients are more likely to be responsive to a treatment, and things like disease duration and vital capacity and ALS-FRS scores and so forth, as we saw with the Adavrum experience, might be considered to reduce the heterogeneity in these early trials, looking for a signal for a response. Something to think about in both of these questions. Thank you.
Thank you. Dr. Van Ness is going to discuss the biological and phenotypic heterogeneity and which factors require stratification, including real-use use, study center, genetic etiology, predictive prognosis, cognitive and behavioral dysfunction, and so on.
So good morning. So as a group, I think we struggle a bit with these issues of either dealing with it in inclusion criteria or through stratification, and probably maybe it even depends upon the phase of the study you're trying to do, how you should deal with this. So I've listed a number of things in regards to stratification. So the first is riluzole is the only drug we know that prolongs survival in ALS. It is standard of care, but for various reasons, not all ALS patients are actually on riluzole. So there is a possibility that if you would select, or if you would not stratify for riluzole use, you would potentially have more riluzole users in one of the arms of your trial, and that could potentially affect survival in one of the treatment arms, or your rate of side effects could be different due to differential percentages in use of riluzole between your treatment arms. So the recommendation would be to stratify for riluzole use. So another one we talked about is clinical trial sites. So this is something we see in all clinical trials in all diseases, that the trial sites themselves are actually a major source of variation in clinical trial data, and that's due to a whole host of things ranging from, I think, cultural differences to differences in healthcare systems, geographical, whatever type of clinic you have. So based on the fact that this is a major source of variation in clinical trial data, we also recommend to stratify for clinical trial site. Then this is what Orla talked about as well. We know ALS is genetically a very heterogeneous disease. Over 20 or even 30 genes are involved at this point. And we know that some of these genes may influence survival. For instance, SOD1A4V is associated with very rapid progression, short survival. In contrast, D90A and SOD1 has a very slow or relatively slow disease course. C9orf and others are associated with faster progression. So that's one thing. The other thing is that specific genetic mutations also have specific pathophysiological mechanisms, right? So we only see the DPRs in C9orf. SOD1 doesn't have TDP43 pathology. So there's different pathophysiological mechanisms associated with different genes. On the other hand, we know that there's large differences between countries and perhaps cultures in approaches to genetic testing. In some countries it's offered to all patients, in some it's hardly done if there is no family history. So this information may not be available up front for all patients when enrolling in a trial, and it may not be feasible financially to obtain this data. So the rationale is unequal distribution across treatment arms of genetic factors that influence survival could affect your outcome, and it seems possible that different genetic subforms might respond differently to a treatment. So our recommendation is that DNA should be collected for all participants in trials, and at the very least, post hoc analysis should be performed based on genetics. and you could consider to stratify at entry, but that may not be feasible. The other recommendation is that I think patients with a certain specific genetic mutation should preferably be enrolled in a gene-targeted trial. So SOD1 patients should preferably be in an antisense oligo trial or something comparable. But in the absence of trials for their specific mutations, I think they should still be eligible for other treatments. So moving on to cognition, it's been shown that FTD and executive dysfunction also negatively influence survival, and both are also associated with poor compliance. So again, unequal distribution of executive dysfunction and FTD across treatment arms could potentially affect trial outcome or dropout rates in trials, and therefore it would make sense to stratify for these issues. So, and then lastly, this also touches on heterogeneity. We know that survival in ALS patients is highly variable. Some patients die within 6 months, whereas others may live over 10 years. So having a different distribution of fast and slow progressors between treatment arms of a trial potentially has the danger of affecting your trial outcome again. Now, over the last few years, a few prediction models have been developed which are quite easy to use, which give a predicted prognosis. So the idea would be that clinical trialists should at least consider using predicted prognosis and try to stratify fast and slow progressors equally over treatment arms of a trial. So those are our recommendations.
Thank you. I think Dr. Miller would like to make a comment.
My concern about all the stratification is that we have this heterogeneity problem, and stratification, that is continuing to have a heterogeneous group of people that we divide into subgroups for analysis, may not be the solution to our heterogeneity problem. I definitely agree that we need to think about how to separate and become more homogeneous, but I would raise the question of whether stratification for all of these separate subtypes is going to really
maybe good idea to get the biostatistician's comments. Eric mentioned before. Eric or someone biostatistically how?
Just a brief comment from why we recommended to do it in this way. So it's not to perform a number of subgroup analyses, it's to balance the number of factors which have an impact on survival. So it's not proposing to do 10 different subgroup analyses, it's balancing the factors that influence. So I can actually back this up with data from a trial. We have trial data where so the number of SOD1 patients was about 5-fold of what you would expect to be in the cohort at the beginning, and then they all ended up in the treatment arm. So I think that's a situation you would want to avoid.
So I do think that the term stratification may be used differently by different people, so it's important to be clear. You're talking about at the randomization, you're going to stratify, you're going to try to match groups and make sure they're balanced by all the types.
Yeah, that's what we mean by this.
But I think the previous speaker meant that you would also do subgroup analysis on all those, which makes the trial, of course, very, very expensive.
So by this point, we mean how to balance things that negatively impact survival in order to not have a skewed outcome. And I think in terms of genetics, that obviously becomes very expensive very quickly if you want to genotype 10 or 20 genes before including someone. So that's the reason why we're recommending to at least take DNA, store it, so you have the possibility of doing post hoc analyses and genotyping those samples if you have funding.
So I'm sorry, let me get my formatting comments in. So the statements in the background shouldn't be labeled as the type. That's only for the rationale. Your rationales are incomplete because you just have inferences, and I actually think you could merge your backgrounds and rationale into one rationale, and you'll be set in that regard. let's see, and then recommendation 4 was in the passive voice, it was an actionable. In recommendation 6, you had the word clinical trialists should consider. Empirical evidence says that nobody knows what that means when you say should consider, so I would try to reword that and get rid of the word consider.
Okay.
Dominic Fee, Medical College of Wisconsin. Next generation genetic testing allows for hundreds to thousands of genes to be assessed with one test. And that is entering the clinical realm. You are talking about still doing the older way of genetic testing where you do either single genes or 10 to maybe 20 gene assessments. Have you actually taken into account the near future, which is the ability to screen for hundreds, if not thousands of genes, and how to take that into consideration on either testing for patients clinically or for research basis?
So we're not making any suggestion on what type of genotyping technique should be used. We're just saying that there's a few genes at this point for which there is evidence, and you can extract that information from whole genome sequencing, or you can get that from a targeted panel if that is the objective. So the thing is, whole genome sequencing will cost you approximately $1,500 per sample at this point, right? You can do a repeat prime PCR for C9 or for far less, right? So we're not saying you should do whole genome sequencing. That is up to the investigator the way he wants to do it. If you store the DNA, you have the option to do that. We're not saying Sanger sequencing is what we're recommending here.
But there's a big difference in terms of the results. If you're only doing 1 gene or 10 genes, your phenotype and the genes that you are looking for is very solid. When you are doing a panel of hundreds of genes, thousands of genes, you run into the risk, or you run in, not the risk, you do run into the situation where mutations are going to be found, and these are going to be in genes that have not been determined to be absolutely causative for ALS or motor neuron disease. And there's actually a big difference between those type of genetic testing in terms of what the results are going to give you and what you need to do to interpret those results.
May I butt in here and say that in view of the tight time that we have, if the questioners could keep their questions very brief, and if the responders also, we'd love to get all the input that we can here, but we are very tight on time.
Thank you. Bob's doing my job for me. Tipu, you're next.
Tipu Siddiq, Northwestern. I think this has been addressed by you and by Ola that we really need to define what genes we're going to test. We must differentiate between association and actual Mendelian or other evidence that is clear and which is agreed across the board as to what constitutes a definitive cause of disease rather than a mere association, which can vary in population and have so many other variables that will just confound the whole issue and make this small population of ALS patients impossible to really interpret unless you have a cure. So I think that before you embark on anything else, that genetic etiological question must be addressed very clearly and very specifically as to what genes are you talking about that are indeed etiological and not just a variant that somebody has reported. You know, I don't think there are 20 or 30 genes at all.
Well, so I would agree with that, but I think the point we're trying to make is that we're trying in large-scale projects such as Project Mind to unravel the genetics of ALS, and that is work ongoing. So by storing DNA, as we learn more and more about the genetics of ALS, we have the possibility to go back to these DNA samples and reanalyze things.
Thank you.
Dave Ennis, Origin Data Sciences. I want to speak to Eric's point about multiple strata, and the more things that we're considering for stratification, the smaller the groups get. And I want to offer up the possibility that tree-based learning systems can utilize all of these parameters and come up with a consensus that uses the relative importance of all the parameters. So I want to offer that tree-based learning systems might be a good way to stratify both for randomization and also stratification for later analysis.
Thank you.
Yeah, so I wanted to touch on the same point. I mean, I think the thing that bothers me, and I actually like the should consider, and I'm going to explain kind of why I do. I don't like the should stratify, because you've got should stratify on way too many things here. There's the real world, and I think there's scientific rationale for why you should stratify on these, but it's not feasible to stratify on all those in a clinical trial. And so I like the language should consider a little better. Maybe it just needs different verbiage, but I think you should stratify on all these in an ideal scientific world, but in any given clinical trial, I think you've got to concede some things, and I think you have to decide which are the critical things you want to stratify randomization on for that trial, and which are you willing to give up on, maybe try to adjust for in the analysis. But I think as written, it's a little too strong, which would say you should stratify on this, you should stratify on this, and you should stratify on this. And I think if you start to slice and dice too many ways in randomization, you're going to end up with a trial that's not going to be feasible, or your groups are going to be too small. So somehow softening the language, whether it should consider or should consider where feasible or something along those lines, I think is going to be important here, because you're going to have to make some concessions.
Thanks.
Hi, Mike Bradburn, University of Sheffield, UK. My experience of stratifying on a clinical trial is exactly the same as that. There's too many strata, not enough patients. The alternative, making a more selective group of patients to study, is that you begin to target a very small subset of a rare disease, and that's too small for pharmaceutical companies to be interested in. That's one of the recurrent themes that's going to come up again and again when we're talking about clinical heterogeneity. I wanted to make one specific point about stratification by riluzole use. You mentioned that usage of riluzole at baseline is an important stratification factor for trials in later stage ALS. NIV usage would also be worth stratifying as well, and that brings you on to that it's not enough to know medication use at baseline, you need to know about it ongoing as well. So I was involved in a trial very recently. One of the criticisms was the NIV usage different between the two arms? We were able to say no, but only because we'd measured that as the trial was ongoing. And I think when you're looking back, you need to have medication usage after randomization as well as at baseline.
Okay, we'll look into that,
So, De Carvalho from Lisbon. Maybe I missed, but there was a mention about progression rate and stratification by progression rate. I wonder if you could use a lead-in period in trials, as has been done in the past, just to select patients based on progression rate. And that could be another thing to be considered as well.
Yeah, so there are a few prediction models that give you some idea of what the predicted prognosis would be. And the lead-in time is one of the factors that is usually a part of those models. So if you use one of these models, you're using that, but there's other things that are also independently of lead-in time predicting prognosis. So you can be more complete than just using that.
John?
I wonder if there's a happy medium between stratification and non-stratification, whereas you design a trial with basically all comers, but you design the trial to prepare for so many patients in each clinical category that you want. So say you have a 500 patient trial and 50 of them have to be flail arm and 50 of them have to be limb onset and 50 of them, and then at the end of the day you have a lot of people in the trial, but then you have enough data to do post hoc analysis, because we really don't know if we pick a specific group that we're actually picking a different group at this point, and maybe that would be a way to do it. I don't know.
Yeah, so I think one of the problems we're running into is that different smaller, bigger studies, phase 2, phase 3, might require different designs and different approaches in terms of inclusion, exclusion, and then subsequent stratification. So I think we as a group might need to rethink the way we've worded this according to what type of study it is.
Thank you. I think it's time to move to the next topic. We're going to move to study design and outcome measures. And I think Eric Sorensen is going to present the first three topics, and we're going to start with primary versus secondary outcome measures.
Yeah, so thank you, Terry. First of all, I'd like to give a big thanks to Hiroshi for organizing this. Earlier in the morning, so earlier this morning we heard about all the people and all the great things everyone else is doing, but Hiroshi didn't talk very much about what he did. And anyone who's ever organized one of these realizes how much effort these are and how much leadership it takes. So thank you, Hiroshi. Secondly, I'd like to thank the members of our group who worked very hard on these documents and recommendations. And I thank you for tolerating my repeated emails for revisions and new drafts. And so thank you very much for all of your help with that. Last week, so this group was led by Len and I, and last week we spoke by phone to kind of divide up. As you notice in the agenda, we're split between this session and the next session. And so we had to decide how we were going to split up the discussion and the talks. And so we decided that we would present in this first section the outcomes that have the strongest evidence, the most experience with, and perhaps the strongest opinions about. And then we would, in the next session after lunch, when Len presents the afternoon data, focus on the outcomes that are maybe not as well developed, maybe not as well validated, and aren't as far along in the process as what some of the things I'll talk about now. So with that in mind, let's see, do I got the, that's not, I think, okay, so this is my first slide here. So just as a way of introduction, I'll just remind everyone what the recommendations were from the previous guidelines. And in the previous set of guidelines published in 1999, there were basically three recommendations. The first recommendation was that all of the outcome measures should be, quote, crisply defined. I thought that was an interesting description. The second recommendation was that the primary endpoint should be either survival or a muscle strength testing. And the survival was defined as tracheostomy-free survival. If you read the appendix, the strength testing was divided in three techniques. One was the MVIC, the maximal voluntary asymmetric contraction, manual muscle testing, and then handheld dynamometry. And at that time, or I should say since that time, there have been a considerable experience with all three of these, and I think MVIC has kind of fallen off in favor because of the expense of the equipment, the space needed, the lack of portability, and the large amount of missing data that occurred with that. The second one, MMT, had a bit of a resurgence. It was portable, it could be done anywhere, it didn't require any special equipment, it was cheap. But it too required a face-to-face visit with the patient and a face-to-face encounter, and it also has had problems with missing data that has complicated analyses which it was used as a primary endpoint. Initially in 1999, the data on handheld dynamometry was really in its infancy at that point, and they really had no specific recommendation on that. I think since that time, handheld dynamometry has really gained a lot of followers and a lot of use, and we've gained a lot more experience with it. And as far as what I can tell, it's really become the preferred method of strength testing in ALS clinical trials. The third recommendation that were in the original guidelines was the inclusion of a quality of life measure. And even though at that time they acknowledged that there were no really well-validated or good quality of life measures for ALS, the importance of quality of life was acknowledged as an important part of ALS a patient perspective. And so with that in mind, I'm going to go ahead and move to where we are now in the work that we did looking at the outcome measures. So just in keeping with the modified Delphi method, the background for this is that reliable endpoints are needed in phase 3 clinical trials. And first of all, the endpoints that are used need to be validated, they need to be repeatable, and they need to be clinically meaningful. In addition to that, they have to be feasible. So you have to be able to recruit enough patients to meet the power for the study. And they have to be designed such that you can minimize the missing data to eliminate statistical problems that come in the analysis after the fact. And so that was really kind of the charge as we looked at each of these endpoints. And I'm going to start with survival. And so the rationale, I think, is people in the audience are probably well experienced with survival's endpoints. Premature, in terms of the rationale using it, premature death occurs in nearly everybody who's afflicted by ALS, and prolonging survival is of primary interest in patients who are suffering from ALS. Survival probably has the most experience of any of the clinical outcome trials and has been used for decades as the outcome measure, as the primary outcome measure in ALS clinical trials. And it has been shown that ALS trials with survival can be done with sufficient power. And as the advantage nowadays in the electronic era, no patients can hide or be lost to follow-up. Every one of our patients enrolled in a clinical trial, we can obtain survival status on them at any point in time. And so that it effectively eliminates any issues with missing data. The drawback of survival, I think, has been alluded to in a few of the other discussions. Because it relies on events, survival events, death events, it requires a period of time for enough events to occur for the groups to separate. And so this requires a longer period of follow-up. And oftentimes, I think you heard earlier, 12 months is sort of a minimum, more commonly 18 months, which is in sort of the peak point of the power for survival analysis is usually chosen. And so in terms of our recommendation is that each of an investigator should include a survival analysis as an outcome measure, either as a primary outcome measure or as a secondary outcome measure, and should be included in any phase 3 clinical study. The second aspect was functional outcome measures. As people know, prolonging survival without impacting or affecting their functional status is a suboptimal outcome and not acceptable by the ALS community. We already have that. If we do tracheostomy, we can do tracheostomy and invasive ventilation, and that will prolong people's survival, but does nothing for their function. And that, as everyone knows, is acceptable to a very small minority of patients with ALS, at least in the United States. So functional outcomes are of major interest to ALS patients. We do have validated instruments. The ALS-FRS was initially developed as a functional scale, and it was then subsequently revised, and so most of our data now is for the revised ALS-FRS. And this has been repeatedly validated and tested in clinical trials and has been used as an outcome measure in clinical trials. And recently, there's been a movement to include it as a primary outcome for clinical trials. The ALS-FRS has been shown to be done with sufficient power. It has been validated to be obtained through a remote assessment, so it does not require a face-to-face visit. And this is a significant advancement in minimizing missing data that has been a problem in other clinical trials. And so it offers a number of advantages. One of the disadvantages that was mentioned, I think, a couple times already, is that it provides some difficulty with an analysis when you have a variable follow-up period of time. For example, most of the time, these scores are changed into a rate of change or a slope score, and so a rate of change or slope score of a change of 3 units over 3 months would be equivalent. Would be looked at as the equivalent of 6 units over 6 months or 12 units over 12 months, but those patients are not exactly equal, and so that does provide some statistical problems when you have a variable follow-up period. Nonetheless, it does allow for a more efficient trial in the sense that trials can be done over a shorter time period when using compared to survival as a primary outcome measure. So the recommendation that we had for using a functional assessment is that the investigator should include the revised ALS-FRS as an outcome measure, and it would be acceptable either as a primary or a secondary outcome measure that could be used in any phase clinical study, phase 1, 2, or 3 of ALS studies. I'm going to move and talk about muscle strength. Muscle strength is probably the hallmark of, or progressive weakness is the hallmark of ALS and worsening of the disease. There have been a number of evaluations and testing designs, methods to test muscle strength, and these have been really pretty well validated. They can be done and can be repeatable, but there is some variability in muscle strength testing. And with many of them, some of them are relatively insensitive to change, particularly early in the disease at the stronger periods of the muscle strength. And that adds some variability, which requires an increased number of patients in terms of power testing. The problem with muscle strength testing is it still requires a face-to-face visit, and as patients get weaker and weaker and travel becomes more limiting, they can no longer make it into the clinic. And so now you're faced with the decision, do you send investigators out to the homes and do their examinations, or do you accept that as missing data? And that becomes missing data, again, becomes a problem with the data analysis and statistical analysis. And so I think that is really the problem with muscle strength testing as it exists today. And so the rationale or the recommendation that we had for this was that investigators may use quantitative strength testing as a secondary outcome measure for an ALS clinical trial, and it would be appropriate for any phase study 1, 2, or 3. If it's going to be done, sufficient patients must be recruited to provide sufficient power and able to interpret the data, and that there must be a plan to minimize missing data as patients become too weak to attend the clinics. Now, the last one that I'm going to talk about is the combined endpoint, and then I'll turn it over and open this up for discussion. The combined endpoint is a composite score that has been gaining momentum, I think, because of the issues that I've talked about with study power, study duration, that kind of thing. I think as Wilson had said earlier, there is no perfect endpoint. And so there's been more interest in these combined endpoints or these composite scores. And so I talked about briefly about the statistical problems in managing variable follow-up, missing data, that kind of thing. And so ALS is not, our discipline is not the only field to have encountered these problems. Other areas have had similar problems, other areas of neurology, other areas of medicine. And some areas have successfully used these combined endpoints in soliciting approval of new drugs. Probably the one that has been most successful, or one that I was aware was most successful, was the use of a combined endpoint in the TPA studies for stroke. In that study, the studies that led to the approval of TPA in the treatment of acute stroke, one of their secondary outcomes was a combined endpoint, which it was a composite score of all 4 of their other endpoints. And the FDA viewed that as a adjunct information and found it fairly compelling, and it contributed to the approval of the drug. And so there's been interest in ALS recently to do a similar kind of composite study. James Berg and his group and colleagues created what's called the Combined Assessment of Functional Survival, which is a composite score that takes into account both survival and their ALS-FRS scores, their functional scores. This is a rank analysis. It's a rank analysis is done, and the patients are ranked on their survival and ranked on their functional score, and then it's combined into a single rank score. It's more or less a statistical manipulation of the data to allow for 2 primary outcome measures between survival and function. This is relatively early in its development. You can see the original papers were just published within the last couple of years, so we don't yet know how it's going to perform in the ALS clinical trial environment. For example, what is if you use something along those lines, how long does a trial need to be, how many patients do we need to recruit, etc. The downside of using the combined score as a primary endpoint is that it does not itself lend to a clinically interpretable data point for the patients. So if there's a benefit on the combined score, how do you take that back and tell patients, well, this is what it means to you to take this drug? I think that's a minor criticism because it's based on clinically important information, being the functional scores and the survival, and of course that data is still available for interpretation and to discuss with the patient. So I think that's a minor issue with that as an endpoint at this point in time. So I guess the recommendation we had about combined endpoints is that the investigator may use the combined survival and functional score, the CAF score developed by James Berg, as a secondary outcome in any clinical trial of any phase. But I do think there is some requirement of some more additional validation, and it's possible that there may be other opportunities for other kinds of combination scores as well. And with that, I will conclude my comments, and I think we can open up for discussion if anyone has any comments.
Hi, Jeremy Shuffman from Phoenix, Arizona. Thanks for a very clear presentation. I have 2 comments. First is that I'm very concerned with formalizing either of those the first two recommendations of making it necessary to include both either survival or ALSFRS as required primary or secondary measures. I think, number one, while the advantages of survival are clear and you summarized them well, the disadvantages are, number one, that it does require not only a longer follow-up, but a huge number of patients. And that I think that if you asked most of the people in this room, I think most people would say that a drug that clearly improves function would be important, whether or not survival was impacted. Secondly, while function is clearly important, the ALSFRS, while validated and used quite a bit, is not the perfect measure. And I think it would be much more preferable to say that some analysis of function should be included in a trial. And you know, a good concrete example of this is that we have a drug that's approved for ALS in the U.S. that was approved neither on the basis of survival nor the ALSFRS, and that's Nudexa, which because of the intent of the drug, a functional measure was designed that would evaluate that drug and was found to be appropriate, and I think that's how we should be thinking. Just the last one, just a relatively small one, is the recommendation number 4. It's probably important to recognize that except for survival, everything else you talked about is a composite endpoint. And that's one of the issues about the ALSFRS, that it's a composite functional endpoint. And because of that, there is the same problem of interpretability as there might be in the CAF score. And strength is a functional endpoint, is a composite endpoint in that you're combining muscles that may be affected, may not be affected, may be affected at some point in the future, but not yet.
Thanks, Jeremy. I just make a comment to that. There's no question that other functional measures or other outcome measures are out there that may be better than what we've got. They haven't come forward yet. They haven't been validated. And this is what we're stuck with today, and this is what the recommendations are based on. There's also no question that survival requires a longer duration, as I mentioned, and often in larger numbers of patients. But I think that as you go to the FDA, if you're going to go to the FDA for a new drug application, and you show that it improves function, I think they're also going to want to see what does it do for survival. So I think you're going to have to look at it and collect the data either way. I guess the question becomes is how do you power your study? What is the study powered for in terms of and that will really depend on which one you choose as your primary outcome.
Just to say that we've already had a drug that has used neither of them that has been accepted by the FDA.
Thank you.
Big, on the same line, I have concerns about using a score like this because, as you said, it has no practical implication, no immediate implications. I just wonder whether you use the two elements like death or reaching a given disability level, and whichever comes first, it's a treatment failure.
Yeah, so you're talking about a time to event analysis or something besides death. I think that would be, I would think that would be fine. I guess you'd have to have the discussions with the agencies and see if that was acceptable, depending on how clearly what the event was. But it also requires a continuous observation period, which is why death is always the one that is chosen.
Yes, Hiroshi from Japan, and I was a PI of the outer high-dose methylcobalamin study, and it took more than 7 years to do the survival. And so that's a phase 2-3 study, and so that's going to be phase 2, and we are going to do phase 3 again. So it takes a long time to do the survival. And so some papers, a couple of papers suggest that, for example, drop of the points of ALSFRS score by 6 or 12 can be the end point. And so I think that approach might be appropriate if you actually did a true survival study at phase 2, and then you can just confirm that using that kind of abbreviated type of endpoint. So what do you think about that?
I think if you look at our recommendations, the inclusion of survival as the primary endpoint was really only phase 3 studies. I think everyone acknowledges that doing that long of a study as a phase 2 or phase 1 is just not practical. and shouldn't be done. And I think for the points that you mentioned, I think either survival or the ALS-FRS as of today are probably both the best options for a disease-modifying trial in ALS.
Okay, thank you.
Thank you.
Marcus Weaver, Switzerland. I have a comment to your recommendation 3. In the background to this question, you mentioned actually that in those studies that applied manual muscle testing, there was rigorous training of evaluators. Right. I'm wondering whether this should go into the recommendation, because if you have untrained evaluators, you create a lot of noise. So perhaps this is something you would like to consider.
Yeah, that's a good point, and I think we could include that into the recommendations.
This is Eric Macklin. I'd like to recommend clearer guidance on how to incorporate mortality in our measures of outcomes that are not mortality, that it'll censor our further observation of those outcomes, and presumably in an informative way. I don't think the CAFS is the appropriate framework, because not only doesn't it provide a clinical measure as you identify, but also you can have a favorable p-value for a CAFS test with improvement in function but not in mortality, or and worsening mortality, or vice versa. So the combination of those two outcomes into a single score precludes evaluating whether we have improvement on both. So I'd recommend instead that we use non-inferiority for survival with potentially very lenient criteria if we are running small early phase studies, and demonstration of improvement on the functional score with an analysis that, as best as we can, accommodate or reduces the potential for residual informativeness of this answering event.
So just a couple comments on what you said. I think you're referring to initially was the survival being tracheostomy-free survival. I think we didn't include that, but I think that is true. We would endorse that. The second comment you said was about the CAFS, and I would agree with that, and that's why I think it has not yet been fully tested how it's going to perform in the clinical trials, which is why we recommend that it may be included and explored. But I don't think the recommendation would be so strongly that it should be included. Oh, just one more point. I think I'm not a statistician, but a true non-inferiority study oftentimes requires thousands of patients and not just a few patients. And so I'm not sure that would be a practical option.
Bjorn Oscarsson, University of California, Davis. My comment is in regards to survival and ventilation also. And today, with the advent of successful non-invasive ventilation, we have a lot of patients ventilating 24/7 non-invasively. Again, making sure that that language is in there also, that we are looking at survival or continuous ventilation.
Yeah, I think this gets to the point of clarifying what do we mean by survival. Tracheostomy-free, 24-hour NIV-free survival. Yeah.
So Eric, this is sort of a question for everybody, and it relates to the nature of the work product that we're gaining toward. And so you've said a couple times, and I think some other speakers have said as well, is that here's where we are today regarding, the status of our outcome measures or the status of our knowledge about risk factors. And so my question is basically, what are we producing? Are we producing a guidance that sort of reflects the state of clinical trial methodology at the moment, or something that's going to guide our thoughts for the future? And to the extent that number 2 is potentially, I think, more important, maybe we shouldn't be thinking in terms of, well, these are the tools we happen to have right now. A recommendation for how we should be thinking about things for the future seems to be, I would guess, more important. And I was wondering if people had comments about that.
That's a philosophical question. I would allow anyone to comment. I guess we've approaching it as what is the current state of the science rather than aspirational.
Maybe Hiroshi or Bob?
Could I make a comment about it? Well, first of all, I think we're going to wait another 16 years till we meet again. It would appear that that's our frequency of meeting. Just kidding. The other groups that are beginning to do trial guidelines that have iterations at a much lower interval, and we need to think about that too. So this is really, we hope, the beginning of a periodic updating that will be done at much closer intervals than has been in the past, and there is precedent for that in other disciplines.
Just a quick question. What about forced vital capacity or slow vital
Yeah,
capacity?
so those are coming in the next session, and Len's going to talk about those when he does them. So I would defer questions on those topics until the afternoon.
Some formatting comments. Sorry, some formatting comments. So first, the format was perfect, so good model. But so our
Thank you.
in there.
Thank you.
So one, I had to come up with something, right? One. So we use 3 helping verbs to indicate the strength of the recommendations: may, should, or must.
Yeah.
And so the default at this stage is should. So you were anticipating what we're going to probably find out in the modified Delphi process, that the may is a may will have a may. But so try not to use may or must. Stick with should, and realize that we'll change that after the modified Delphi process.
I think in the documents as they appear in the paper, it says must, and we changed it here to should. So thank you.
So may I comment to comment by Jeremy's? And if you remember a slide I showed, I think it's a very difficult balance. We should not limit the clinical trials. I think this is more facilitatory and expanding. And so language, I think you have to be very carefully use wordsmith, almost wordsmith. So you can't say, oh, everything is neutralized, everything is okay kind of recommendation, but at the same time cannot be too restricted. So language has to be equally facilitatory and doing things right, more expanding clinical trials. That's what I hope this meeting will do. I'm not sure I'm saying right things, but that's what I hope it will see.
Yeah, I mean, I usually don't, but I must say that I probably agree with Jeremy that I think it is good to identify certain points that should be improved, and I think we can identify several limitations of the ALS-FRS score, and we probably need a better functional scale. And I mean, it would be probably not good if that's not written anywhere. Is that if that would not be one of our conclusions from this meeting, that we should work on that. So back up from Europe for Jeremy.
Thank you, Leonard.
So a couple of things, if I could make another comment. The first is that the morning has been rich in terms of hearing the presenters and all of the deliberations of the groups, and coming close to the proper formatting and having a document that pertains to some of the most important issues in improving our clinical trials. So the goal is that any point that you didn't get to make or that you feel is important to the group should be put into an email today or tomorrow and get it to the group leader, because tomorrow and subsequent days they'll be meeting again. They will reflect upon the feedback that they've gotten here today, and also the feedback that you give to the group leaders about things that you feel either haven't come up or that you feel very strongly about, so they can take that into account. I was asked secondarily, and each of the groups are asked to look at the old consensus guidelines in one or two respects. And I wonder if I could ask you to turn in your program booklets to page 83. There are 2 points in the old consensus guidelines that are not really being addressed in the discussions that we've had so far. One is number 10. This relates to how to release the results of the trial. And it's very short. I'll read it to you. And then I'll ask whether you think this is okay, or whether we should get rid of it, or we need to substantively revise it. It says, the manner in which information is released during and after trial should be an integral part of the protocol. It's recommended that no efficacy results be released until peer review publication is imminent, other than at scientific meetings. We recognize there are legal constraints on the release of trial results when a trial is terminated early, because of futility or strong evidence of efficacy. Such release of information should take place only after release of information to trialists and to the patients enrolled in this trial.
Bob, sorry. So we finished Eric Sorensen's presentation, right? Sorry, we finished his presentation, right?
Eric, do you agree with that?
Okay, sorry.
So Eric agrees, Hiroshi. I think we're.
We should have given a round of applause to Eric.
Thank you, Eric.
I'm wondering whether anyone has any comment or question about that release. I see a couple of hands going up. Andy and then Wally.
So Bob, I do, and I certainly agree that the investigators and even perhaps the patients, if it can be expeditiously done, need to know the results. But if the study has been sponsored by a company, then it may be material information, and we can't sit on it that long legally. It needs to be disclosed at least in some top-line degree. You know, if it's especially if it's either very, very positive or very, very negative, so that the so-called reasonable person would make decisions about whether to buy or sell the company's shares. Generally, legal advice that we've been given is no longer than a week can you be in possession of that data and not communicate it, and the less time you have it without communicating it, the better. So
let me ask you a question, Andy. Would you be comfortable with still, obviously the legal issue trumps all, but in that short interval of giving the sites the chance to share the information with their patients, and also convening, let's say, a webinar or a home conference so that the trialists could hear the results, react to it, give feedback about the release of the information and so forth. That was the spirit of this.
Well, certainly I think I began saying that the investigators should hear it first, but that interval between when the investigators hear it and the public release is made needs to be as short as possible, because if it leaks out, and it can and has, before it's been formally disclosed, it could be a world of hurt for the sponsor.
So it would be great if I could get some draft comments from you about this, and Wally.
So I just wanted to add to that point. Back in the BDNF trial days, we had that agreement from the sponsor that in fact it would be revealed to the investigators prior to any more public announcement. And we were cut off by the Security and Exchange Commission regulations that they said indeed they could not do what they had promised to do because they were a small company, and the release of this information would in fact have a major effect upon share prices and so forth. And so we may say in this recommendation 10 that that's what we want to have achieved, but it isn't necessarily what in fact in the end is going to be possible, particularly for the pharmaceutical sponsored things. But the other comment I wanted to make is that we ought to come up with some recommendation that prevents the release by pharmaceutical companies, small that they be, of apparently suggestive positive results that then in fact are used by that company to enhance their investability. It is something that I think we've seen in this last few years to be really an ethical problem for us as investigators, and somehow we've got to get to prevent that.
I appreciate your making that point, Wally. I see a number of experienced clinicians nodding agreement about that, and it would be really great if you could draft something. Wally, if you could give me a draft of a few sentences about that, that would be great. Any other comments then about this particular point of dissemination?
Yeah, just a quick thought. I mean, we've talked about the kind of company side of things. I think in academic trials, there's also the issue of, I mean, they usually don't have the legal requirements, and you're not bound, but you might tell investigators and patients who participate in the trial about results. What's different between today's world and the world of the prior guidelines is that social media makes it so that as soon as you tell patients everybody knows. And so they know so you either are going to release the results to the world via the people who heard it on a webinar, or you're going to release the results to the world via a broad, some sort of broad dissemination. And I think we just have to take that into account. The order of operations may be the same, but the timing between them may have to be rethought.
Thank you. Good point. Dan?
I have another point. Stan Moore, California Pacific. I didn't see any recommendation there about data sharing. Speaking as a statistician, this is very useful in designing future trials. And we do have a PROACT database, which I think will accept results of clinical trials in any format. So I think maybe that's something we want to add, because it's already established that we have a repository for this, and it would be useful in the future.
Thank you, Dan. A very good point also. I totally agree about that. The other issue from the previous guidelines that I wanted to just bring to your attention is about publication. And point number 20, which is on page 84. Yes? Sorry?
Yeah, but we discuss, because we have to, as a group, we have to discuss this point. We have a slide on.
Oh, great. Is that coming? Great. I apologize.
So basically, the group agree that we can agree about this point, that doesn't require major changes. Obviously, it's open to discussion. We feel, yeah, we feel that is enough comfortable, probably, the publication committee requirement as was defined in the previous guidelines.
Question 20. Yes.
This is question 20.
Yes. Thank you very much. Anybody have any issues or questions or comments about the issue of publication? Thank you for taking the time.
Are there any more comments that anyone would like to make in this morning's discussions? I want to thank all of the speakers and committees for their hard work, and Hiroshi again for putting this together. And I think that it's lunchtime. Thanks again to all the speakers.
So lunch will be ready noon, so in few minutes, and we are ahead of time a little bit. That's good. So we're going to reconvene at 1 p.m. So at least you have 1 hour. Enjoy lunch. We'll see you.
May I make just one last comment? For the group leaders this morning, there was some overlap and repetition. For example, in gene testing and so forth, and the group leaders might want to confer about how to eliminate some of the repetition. Thank you. No.